goes, we will give a notification if time is kind of running up for the speaker. And feel free to type your questions in the chat. We're gonna check it regularly. So let's get going. Our first speaker for today is Zoe Finfrock. She's our staff scientist with a structural biology center at Argonne National Lab. She got her PhD in environmental physics, and her research interests have been focused on developing novel X-ray techniques in a wide variety of applications, especially in imaging and spectroscopy. Today, she's going to talk about her effort in developing a new user program at Advanced Photon Source. Please go ahead, Zoe.
Thank you, Olga. So can you see my slide?
Yes.
Okay. Thank you, Olga, for the introduction, and welcome back to the second day of the workshop. Today, I'm going to talk about a new user program, a new initiative that we're working on to develop at the APS prepare for the APS after the upgrade. Okay, and you have learned a lot about an APS upgrade, so I'm not going to repeat all the information, but just want to highlight some of the key features of the APS upgrade. We will still have the high energy, and then we will get a high brightness and high coherence. All these unique characteristic of the APS is actually position the APS to be one of the best imaging synchrotron source in the world. So to fully take advantage of the APS upgrades, or we are developing a program try to promote biological environment user community, explore and access to the synchrotron user facility. So the program, we call the program the Light. It's essentially integrated a platform and utilize multiple X-ray technique and the various beamline at the APS, and to help address some of the challenges in biological and environmental research. So the Light program will take advantage of the imaging capability. So our program is essentially the X-ray beamline is a center pillar of the program. And within this program, our user will have access to protein MX, protein crystallography capability, and full field X-ray tomography capability, and X-ray microscopy. On top of that, those are like the three main focus of the program. But on top of that, we will also have dedicated limited effort into X-ray spectroscopy and small angle scattering. And besides of the X-ray capability and beamlines, Light will also leverage the existing infrastructure at Argonne site, such as the lab resource and supercomputing, and to help support our user X-ray experiments. And we have the advanced protein characterization facility, and also has a microfluidic lab, and then we're also in partner with the APS cryo lab, and also there is on-site Argonne has a fully equipped biogeochemistry lab. It's just about a half-mile away from the APS. And we'll also work with the Center for Nanomaterial. Actually, the CNM is a user facility themselves. In our case, we will use as for like sample preparation and sample preparation, screening, and some like a novel sample like a chamber devices designing, etc. Like I said in the beginning, and the APS will be one of the best synchrotron for imaging technique. And you have heard a lot about microscopy and tachography and X-ray full field tomography imaging technique yesterday, and I think you're going to hear a lot more today. So I'm not going to go talk about those capability, and I'm just going to highlight a couple other resources and capability that you can utilize to imaging and biological system and the environmental system. So one, the imaging and protein crystallography, basically you can visualize the structure information at atomic level, and with the MX. And so for this type of a study, we can study we can look at a single crystal. We can also do the in-situ study with like microfluidics system delivering, and we can also do serial crystallographer and the time resolved protein crystallographer. And then those experiments can be done both at room temperature and cryo temperature. And talk about X-ray microscopy, and I want to mention there's another unique capability at the APS, is called confocal X-ray microscopy. Basically, you can put optics on your detector, and which allows in visual, actually, like you can put optics in front of your detector, and for study like bulk sample, and the focusing optics in front of the detector will give you depth sensitivity and bulk materials. And so here I just show an example. Is that it's a section of a bone, and this is a study of it's a lead toxicity, lead uptaking in bone, and those are I think the sample is from like the Franklin expedition, and then and the soldier died during that, and scientists speculated that it would be the death was caused by lead toxicity. And so, and the scientists got some of those bone sections from the burial site of the expedition, and those bone samples were pretty preserved. So it's really hard to get a skin section, as most of you, the X-ray microscopy knows that for a lot of the microscopy study, you require a thin section sample. So in this case, the thin section is not actually possible. So, and so we did a series of study like with the bone, with these bone sections, and without confocal, and with confocal use polycapillary. As with confocal, we use a collimating channel array that's actually developed by a group at CHEST led by Arthur Wall. And as you can see that with the conventional XRF and in bulk sample, you don't really see much of structure information. With polycapillary, you can see that the resolution, the depth resolution is energy dependent. So with a high Z element, you can see some structure information, but when you go down to a lower energy element like a calcium, and then you don't really see much of a structure information. But with the collimating channel array, you can see there's the depth sensitivity and the depth resolution is consistent throughout the energy range. And with confocal, you can also do spectroscopy, like a spatially resolved spectroscopy. Also, there's another advantage of using confocal is the background reduction. This is This is iron spectroscopy from a pigeon eggshell. So the red is collected, the red signal is collected from a single channel vortex detector and combined with polycapillary. And the blue is a 4-channel vortex detector, 4 element. As you can see, with confocal, you are winning by just reject the background noise, and the signal to noise ratio is quite significant. And I just want to show a couple other applications in confocal microscopy. This is a study of gold non-rod in plant leaf, and it's a collaboration with Ken Kaminer. And it's another is a selenium toxicity in zebrafish. And we also did some study on onion peel about calcium distribution. And this is just a redox map. You can do redox map at chemical speciation. It's a biochar. It's actually using this study is using biochar to clean up the chromium contamination in the mine field. And the other technique I want to highlight is a high energy resolution fluorescence detector. It's called a HERFD. And most of the people use the HERFD, they actually use for this spectral sharpening applications. So in our case, we use to study a Roman coin, and it's a gold coin, and people want to know the platinum distribution in the gold coin. Apparently, the platinum signature can tell you where is the gold source coming from. So as you can see, we can get in the spatial distribution of gold, platinum particles in the gold, and then we can also do this spectroscopy in getting the chemical information about the platinum. And the other for spectroscopy, you can use X-ray emission, and to actually, this is the mini XES spectrometers developed by a group at University of Washington led by Professor Jerry Seiler. So you can get a single shot to get a full spectrum of element, and then in doing this speciation mapping. And the other technique I want to mention is the X-ray Raman. And so basically you can use a hard X-ray to probing low Z element spectroscopy. So, and this is one of the upgrade feature capability. And you can do carbon imaging and carbon spectroscopy with X-ray Raman technique. The other capability for imaging I want to mention is sex and wax. Basically, sex and wax can help you study like particle size, structure, and shape, and etc. And the sex wax beamline and the APS, and they are really pushing the boundary for this technique, and they can study a particle from atomic level all the way to like a single cell, like a microbe and bacteria. And we have worked with this group to study some aerosol particle and determine the size distribution. As I mentioned, within our program, we have several facility and lab resources to provide for our user to ensure their success in the X-ray experiment. One, this facility is called the Advanced Protein Characterization Facility, is directly attached to the APS storage ring. And in the APCF, we have a full protein production and characterization pipeline. And you can, from cloning, sequencing, and crystallization, and deliver them to the beamline to get the structure information. Other lab resources, the microfluidics lab, and microfluidics is you heard a lot from Georgia talk yesterday, and you can use to handle a smaller volume of sample. You can use it for protein crystal crystallization, and you can also develop those like micro chamber to grow plants and monitoring bacteria colony in plant root. And the other lab resources, the APS the cryo lab, is specialized to prepare cryogenic biological specimens. And you can, there's various instrumentation you can use for section preparation and sample screening, etc. And another facility is the biogeochemistry lab in Argonne, which led by Dr. Ken Kemner and Judy Jastrow. We have used, they have a large giant walk-in freezer that we can use for some sample preparation for permafrost, etc. And we have worked with the CNM and in like for sample screening. And we did some like the microfluidic chip device design. We also work with the CNM for like nanoparticle labeling for protein for like imaging with XRF technique. So having those lab resources is essential to our user community. And then there's just, I just want to show a couple of the example that we did through our pilot activity to develop this program. And so for how to work with us, and so Eberlight is basically a virtual CAD beamline. So we're acting just like all the other beamlines. You can apply through the user general user proposal, but within Eberlight, if you choose us, and your proposal will not be competing with the rest of the APS general users. So as I mentioned, the program is focusing on promoting the biological and environmental research community. And we are also working with other DOE labs, and we just joined the FICOS program and this year. And so you can also submit the FICOS proposal and to work with us jointly with like GGI and ANSO and it said take a village to raise a child, but it sure take a whole bigger, larger village to developing this program. I just want to thank you everybody on this team. I probably missed some names, and we also want to thank our user that to work with us for this pilot activity. And thank you for your attention. If you have any question, I'll be happy to take it. I'm still, okay, I stop sharing, is that right? Am I stop sharing? Yes, okay.
Yes, you stopped sharing.
Okay, so, okay, and I think it's right on time. Do we move on to the next speaker? If nobody has any question.
I guess feel free to email to Zoo if you have any questions regarding application to this program.
Okay, so our next speaker is Dr. Brandy Stewart. Go. So Dr. Stewart is currently a visiting scientist at the Argonne National Laboratory and studying colloidal transport. As an environmental engineer, she is passionate about the interaction of contaminants in the environmental system and design solutions to minimize our impact on the nature setting. So today her talk is focusing on our XRF study in mechanism of chromium removal from industrial storm water using peat medium, sorry. Go ahead.
Great. Hopefully everyone can see my slides.
Yes.
Okay, well, thank you so much for the invitation and opportunity to speak to you today about some of the research that we did. This project, unfortunately, like many, sort of straddled the pandemic. So we began this before and then had some slowdowns with, we had our samples, but slowdowns with beamtime access, et cetera, in finishing it up. But luckily it's wrapped up now. So I'm going to talk to you a little bit about our site and our sort of unique and difficult sampling strategy here, and then some of the synchrotron-based work that we did to understand and interrogate the system. So this is Diamond Chrome field site, which is in Michigan, outside Detroit. And what they do at Diamond Chrome is a chrome plating facility. So metal parts come in here unfinished and unpolished, and then they get this chrome plating and they leave like this middle picture. But what you can see up here is that they also have a water treatment system, and they have this actually not for their chromium waste discharge, but it's actually for their stormwater, because they have so much dust around the factory and on the roofs and in the parking lots that they actually have to collect all their stormwater. They have a big storage tank, and then intermittently they run it through this gravity fed treatment system so that they can then discharge their stormwater once they are underneath the discharge, in which case for chromium, it's the drinking water limit. This is a little bit of a closer look. So there's 7 tanks ran in parallel, and the water comes in the top from the big tank where they store it, drains through the medium in the tank and then out the bottom. And you can see just looking around that this is a very dirty industrial setting, hence all of the dust and chromium, zinc, all sorts of metals. This is an aerial shot of the facility, and I just sort of want to point out again a lot of pavement and then all of these roofs, and so we've got metal laden water sort of running off a lot of surfaces. And this is their tank where they collect, and then if they're in a rainy phase and it fills up, then they start running the system, but it might sit idle for days or weeks until they're running out of storage, and then they operate the system. Little bit of a cartoon image of what happens. Here's the reservoir tank would be over here. So then they pump the storm water in. More on the mixer in a minute, but they have an inline mixer where they're adding iron into the system. Then it comes into one of the 7 tanks and then out the discharge area. And in here is where we get to the other part of our project. These tanks are filled with a commercially available peat, as in peat from the ground, sorptive material. And what we were trying to do here, spoiler alert, the system was working very, very well long before we arrived, but they had no idea why. And it's not actually the chrome plating facility, but it's the people that manufacture and sell the peat product. They're very motivated and interested in understanding why this works and what the capacity for metals as well as inorganics are so that they can market their media. So just a reminder here, we're largely concerned with two oxidation states of chromium, chromium-3 and chromium-6. And chromium-3 is generally thought of as less mobile, often solid. We think of it as benign. For a long time, we thought it was essential for mammals. It seems now that that may not actually be true, but we'll think of that as the safe chromium. And then we have chromium-6. That is our very strong oxidant. This is the kind of chromium, unfortunately, that is used in all of these manufacturing processes here. And this is our less desirable form of chromium. So this is what American peat technology does. They own land that has these peat bogs. They harvest it, and then it goes through a slow homogenization, kind of cleaning out of big sticks and rocks, and it's dried, and then it's kind of ground back up to be a little bit more uniform and a little bit more calcitrant, and then it's in these reactors. So our approach was to go in, sample these reactors. We tried to core the reactors so we could get a sense of what was going on depth-wise as well as radially in the tanks. And then we're going to use a variety of spectroscopic and microscopic techniques at different scales to try to understand more what's going on. Just an aerial shot down to the factory floor. Here's the chromium baths where they would actually do part of the chrome plating. And those are filled with chromium-6. So this is looking down into the reactor and just showing that we tried to sample sort of from the center and then closer to the edge and in the middle. And when the water comes in, it sort of comes in and bounces off a spill plate so that they try not to have a steady stream of water sort of entering straight down into the middle of the reactor. I would say that works somewhat effectively. And this is just us trying to sample the reactors. The ceiling is, I don't know, 2 feet from the top of these tanks, so not a particularly easy place to sample. And you can see in this picture here that when it goes through the heat tempering process, we lose a lot of the coagulative properties also in this media. So it was very hard to use a traditional core. We couldn't because when we tried to do that, it would just fall back out the bottom. So we came up with a plan to actually vacuum the cores up into a piece of PVC pipe and then extrude them. So that's what my coworkers are doing here. If we take a little bit of a closer look at the peat, this is just a light microscopy image of some peat mounted on a slide. This is what it looks like when it comes out of the reactor. And then we can see with our SEM image here that it's kind of got this platy structure, which we expect to see from a high organic carbon material. And then we start to just sort of see this layered effect indicating maybe not some internal porosity, but also just fairly high surface area, which we're not surprised to see given its ability to retain all of these metals from the water. All that I'm showing here, especially if we look over on the right, is that the peat does break down over time. So these samples were from, had been in the reactor between 3.5 and 4 years versus the unreacted samples. So it's just getting a little smaller and breaking down, which you can tell because when you rub it together, it's not super hard for you to kind of fracture it a little bit. I won't spend too long on this, but I just want to point out here that the chromium comes in. Presumably, it's largely chromium-6 from the manufacturing processes. And then there's this iron-2 in the form of ferrous sulfate pre-treatment step. And that's designed to ensure reduction from chrome-6 to chrome-3 before entering the reactor. And they do this because they're just so worried about chromium-6 floating around down through the reactor and out. And we can see that it is working effectively because we're well over the drinking water limit at the inlet and then down below at the outlet. We were able to sample twice. After 3.5 years, we went in and then sampled, and then they took the media out and replaced it with fresh media. And then we went back and were able to do a second sampling with new material that had been in a year. So we were taking a look at the total chromium to get a sense of what we were dealing with to begin with in both. So we can see here that chromium is accumulating the longer it stays in the reactor, unsurprisingly. And we can see when we do an oxalic acid extraction, which is particularly designed, or not designed, but it's effective at only pulling out chromium hydroxide in the system. And we do get some, but when TCLP was performed, which is an EPA method designed to see if something was safe for a landfill, no chromium was released. So initially that was, puzzling to the people at the factory because they knew the chromium was going somewhere. Just a little bit of TEM work to kind of try to get a sense of where the chromium is and how it's interacting with the peat. We can see here, iron is all over our system. We'll also see that in the synchrotron data. There's iron originally, in the peat soil, but then they're dumping in excess with that pre-treatment step. So now what we're looking at is a polished thin section, and this is an XRF image. And these are our actual these are our actual peat particles with chromium associated with them. So everywhere that's bright here is where chromium is actually forming kind of a rind around the peat particles. We get little individual bits of chromium, and then we also get it around the edges. And then if we take a look at what's happening with other metals, we see that, as I mentioned a moment ago, lots of iron in the system. We see zinc. We also, not shown here, we see cadmium. We've got places certainly indicated by purple here where we have chromium and zinc co-located. We've got these very pronounced chromium rinds that are forming, like we saw in the single element image. We've also got little bits of chromium. And here's a closer look just at that prize particle here that's in the yellow circle. And we certainly see sort of this accumulation around the edge. The other thing we were able to take a look at is what was happening over time. And so on the right, this is also from the 3.5-year sample. And on the right here, we see that this rind around the edge is getting fairly uniform. It's connected, and it looks much thicker than when we took a look at a sample that had only been in the tank for a year. We can see chromium is finding the edge of the particle, but less. So clearly it's accumulating over time. And then we went in and did point stains at various locations in these samples, and we started to see more than two, but sort of two categories. At first we were wondering, we thought somehow we had contaminated our sample or messed this up because we have these areas that look like the chromium foil. And then we have these areas over here that look like sometimes chromium hydroxide, which would be this more green spectra. And then we've also just got chromium-3 adsorbed onto organic surfaces. But it turns out what we realized this was this is actually little pieces of metal filings that are around the factory, from preparing the chromium baths that are finding their way into the reactor. So when we compile everything, first we'll take a look at the chromium data. And on the y-axis here are, from different places in the reactor and different time points. This 3M is from our 1 year, and everything else is from our 3.5 year. We start to see in the later time point that we have more variation in the spectra, and we certainly also have more chromium. And we can also look at what's going on with the iron. And then when we bin this and kind of try to separate it into large categories of chromium to understand kind of the overall behavior, we see that we have appreciable amounts of chromium and iron hydroxides. We've got our chromium 0. We have a large amount of chromium, organic chromium, and then we also have mixed chromium and iron pellets. And what those are from a spectra that we borrowed from some of our collaborators at the ALS, and they had done a study where they had chromium and iron that was precipitated in the presence of cellular material. So this is another kind of organic representation, but it's got slightly different characteristics in the spectra than straight chromium-3 adsorbed on to organic. And we're not talking about iron too much today, but I just want to point out that they really are adding a lot of excess iron too, and it's still kicking around the system. And that's why we see that big fraction of iron too. And then we were able to take our same thin sections and then make a chemical map here. And I want to point out that we see absolutely no chromium-6 anywhere in any spectra. Unsurprisingly, we see predominantly chromium-3. Certainly, they ensure that with the pre-treatment step, and then we see it accumulating in the peat. And then we also have our little areas, and they generally aren't in the rinds. These areas of chromium-3 are generally standalone particles that are not associated with the peat. Those are the little pieces of metal that I was mentioning. So what's going on is that we have appreciable chromium sequestered in the peat media. There's certainly some preferential flow or channeling. We suggested to them that they could improve this process. They do fluff it from the bottom occasionally, but they don't have a great routine for that. And they're certainly not approaching it very systematically. And so what's happening is all of a small fraction of the peat is seeing all of the chromium and other metals. So it looks to them like their media is really spent sooner than it is. So they could do a better job sort of regenerating and ensuring that all of the sorbent gets used. Our predominant species are these chromium-0 metal pieces or hotspots. And then we have chromium-3 rinds that are a combination of chromium hydroxide or mixed chromium iron hydroxides. And then in addition to that, we have chromium-3 adsorbed to the surface of the peat particles. And an additional question that we wanted to answer was, what would happen if chromium-6 entered the reactor? Because it was interesting to us that we didn't see any chromium-6 in any spectra ever. So then we ran a sort of a side series of experiments that I did not show today, where I took other peat, like other media that the company gave me, and I set up some incubations where I just added chromium-6 to them. And in under 24 hours, there was no chromium-6 left in the aqueous phase or the solid phase. And at first this was a little puzzling, and then we realized, well, what's likely going on is that the chromium-6 is such a strong oxidant that it's interacting with the functional groups on the surface of the peat and instantly reducing. So it's really not necessary that they add all of this excess iron to in the beginning. They could certainly save material and money with that, cutting down on the iron. And with that, I'm happy to take any questions.
Great talk. And you made a great time too. Okay, do we have any questions? I just realized I was trying to put my finger up and tell you like there's 2 minutes left, but I don't think you can see me. Okay, do we have any questions? Somebody has a hand up, Su Chen.
Hi, Bernie. This is Sichen from the APS. Very nice presentation. I've learned a lot about this topic myself. I have a question about your speciation measurement. I wonder about when you take this measurement, whether radiation damage is a concern. So basically, when the X-ray photon react with the sample, do you think there's a potential impact from the X-ray photon itself?
We did think so, but that we did check for beam damage by running multiple scans in multiple areas, and we never saw any changes. And we were not, because the system was open to general oxidation, in the system, we tried to preserve the redox state, but we weren't overly worried about that because it's open to oxygen to begin with. But we were worried certainly about beam oxidation or reduction, but we didn't see any evidence.
Okay, great, thank you.
Yeah.
Any other questions?
I might have missed it, but what happens to the peat media after it's been used?
Oh, I did not mention that. So they're on the second batch of it now. And because it passed that TCLP test, which is the sort of main EPA determinant for whether something needs to be handled as special waste or hazardous waste, or can go to the landfill. So because it's tied up and bound in chromium hydroxides, and that organic acid extraction doesn't pull any off, they were actually able to discharge it to the landfill. But it's certainly staying in there for several years before that.
Oh, okay. Thanks.
Yeah, thank you.
Okay, thank you. And maybe you can stop sharing. We'll move on to the next speaker. Maybe you can stay after the session. Then if anybody else have a question, then we can discuss it later. Okay. Thank you.
Sounds good.
Our next speaker is Laura. Can you share your screen? So Dr. Laura Sullivan is a postdoc in trace metal biogeochemistry at Beetle Lab for Ocean Science in East Booth Bay, Maine. She measured the cellular stoichiometry of marine phytoplankton and the trace metal content of co-occurring particulate matter to understand the relationship between environmental nutrient availability and the nutrient content of phytoplankton. And she received her PhD in chemistry from UC Berkeley. Today, her talk is synchrotron X-ray fluorescence spectroscopy to measure trace element stoichiometry of individual plankton cells. Go ahead, Laura.
Thanks for the invitation to present in this workshop. As mentioned, I study both the particles in the ocean and the subset that is phytoplankton, and today I'll be focusing on the phytoplankton.
Very simple.
Let's see if I can, okay, there we go. So spend the first portion of the talk giving some background on the context for our research, and then I'd really like to focus on how we collect and prepare our samples, and then how that determines what some of the limiting factors in our analyses at APS and what we hope for the upgrade. So Ken's opening talk yesterday included this diagram of carbon cycling, which I was pleased to see. And I've highlighted here in the larger font some of the key reservoirs of carbon. On the right side, we see that the deep ocean actually is the single largest carbon reservoir on the planet, with almost 50 times more carbon in the deep ocean than in the atmosphere. The surface ocean, meanwhile, has a similar amount of carbon as the atmosphere. And so combined, the marine carbon cycle is a key control on atmospheric CO2 concentrations. Carbon makes its way to the deep ocean from the surface ocean by way of the biological carbon pump. So here, dissolved carbon is taken up by phytoplankton through photosynthesis. And in that organic form, it can sink as dead cells, or these phytoplankton can be consumed and excreted, and that detritus can sink to the deep ocean. So phytoplankton are the key player in moving atmospheric CO2 into the deep ocean through this pump. And my research focuses on understanding the controls on phytoplankton growth. So phytoplankton are floating algae and cyanobacteria. They require the usual macronutrients to make their biomolecules, carbon, phosphorus, and nitrogen. And in order to acquire these elements, they need trace metals as often as cofactors in enzymes or in transporters. Because of all the interplays between different trace metals and nutrient availability, we often collaborate with omics groups to understand some of the cellular pathways that are being expressed, to put our measurements in context. And we really value the multi-element analysis capabilities of synchrotron measurements, because then we can understand how possible substitutions or interplays are happening. So if we look just at iron, which I've highlighted in red, it's needed in photosynthesis and nitrate acquisition, among other cellular processes. But iron is not very soluble in seawater. And so in a third of the ocean, iron is the limiting nutrient that controls phytoplankton growth. This map shows nitrate concentrations in the background, and then the center of each circle, the color indicates the primary limiting nutrient, and the boundary of the circle indicates a secondary limiting nutrient if there is one. So in the Southern Ocean, in the Bering Sea, and then the Eastern Tropical Pacific, iron is the primary limiting nutrient and often the sole limiting nutrient. I'm interested in how phytoplankton groups differ in their responses to these nutrient conditions, light conditions, temperature conditions across the globe. And most of my work is from environmental samples, although I do a little bit of culture. So I want to share a few key ideas that motivate our current research and then discuss how those determine our sampling needs. This is a study from about 30 years ago where a variety of coastal phytoplankton strains were cultured under varying iron conditions. And it was shown that as iron availability increases, phytoplankton take up more iron. So we call this luxury uptake. Notably, there are a few strains that don't exhibit that trend. And that's one of the interesting things about looking at the open ocean samples. This range, so we have about 3 to almost 100 micromoles of iron per mole of carbon in a cell in these culture samples. And that range is also seen in environmental samples. So this was a study from the Eastern Tropical Pacific, from the coast over here on the right, out to the open ocean near Tahiti. And you can see that the range is similar, but these two colors are two different large taxonomic groups. So centric diatoms in green, and then red are small autotrophic flagellates. And these respond pretty differently to the changing conditions over this transect. That's one of the research areas that we're really focusing on now, is teasing apart what makes different taxa respond differently. These lines are model predictions, and you can see that models are still pretty poor at predicting some of the iron quotas in certain regions, reflecting that we really don't understand all of the controls still on stoichiometry. We also see this, I'll go back here a sec, you can see that even among these cells, there's a, what is this, twofold difference just from in this error bar. There's a pretty large range in stoichiometry within a coarse taxonomic group. And that's a variability that I've seen in other studies. This was one where I was looking at the smallest picoeukaryotes that we can measure, less than 1 to 3 micron cells. And these are slightly larger cells, 3 to 10 microns. And in both of these groups, we have way more variability within the group than between the groups. So these two key questions I've mentioned, how are environment and cellular stoichiometry related? And then how do taxa differ, have been really directing our research recently. As I mentioned, these samples are from the open ocean. So we collect these by going out on research vessels with specialized equipment for trace metal sampling. In this center picture, you can see a lot of rust on a ship deck. And so that's a problem if you're trying to collect cells in a trace metal clean manner. To get around that, we bring special bottles that are all plastic. There are no metal parts on these. We use a non-metallic wire to lower this sampling rosette into the ocean. Once we bring water up, we bring these bottles into a trace metal clean van. So this is a van like this. This particular van is not actually the trace metal one. The trace metal was, you can see the corner of it here on the starboard side. But inside we have HEPA filters up top for our air intake. And this is, again, avoiding any metal inside. We take water from these bottles and centrifuge it. So first we fix our cells with glutaraldehyde to prevent any leakage from those cells through subsequent processing. We then put our silicon nitride substrate for the synchrotron analysis on an araldite base, just a resin that we've cast to fit into a centrifuge tube so that the silicon nitride will sit flat. And then we fill the tube with our seawater and centrifuge that down. So we end up with all the particles that were in the water column concentrated onto that silicon nitride window. Not all of that is cells. A lot of that is dead cells or pieces of other organisms or just particles. And so we then go through and use epifluorescence microscopy, looking for chlorophyll A signals to identify living cells, living at the time that we collected and fixed them. Once we're at the synchrotron at 2 IDE, we do a quick fly scan and look for the macronutrients, phosphorus and sulfur, to confirm our cell coordinates. And then we set up our long step scan to that we'll use to quantify the metals. These scans take about an hour per cell. We usually adjust our dwell time and our pixels accordingly to get to try to do most of these in about an hour to enable us to get through a decent amount of cells. But that ends up being one of the limiting factors for what we can analyze. So if you look at the size range that phytoplankton cover, they cover 4 orders of magnitude, from the smallest, less than 1 micron cells up to these trichodesmium colonies that can be 1,000 microns. The cell I just showed is an example. It's a Cosconodiscus, but it happens to be smaller. It's in this sweet spot for synchrotron analysis at 2 IDE right now. And we measure a large variety of taxonomic diversity in this range, but we're still missing a large portion of the naturally occurring phytoplankton populations. So we've tried pushing the lower limit on this. This is Osteococcus lucimerinus, which is a eukaryote, but is in this, it's the smallest known eukaryotic phytoplankton. So it's in this size range with these cyanobacteria around 1 to 2 micron diameter. And you can see the resolution with a 0.4 micron step is it's a lot coarser than when we're doing these large cells. So it becomes less useful and a little bit fuzzier to distinguish our cell boundaries. That's pretty limiting, actually, because if we look at the abundance of these different phytoplankton groups in the ocean, these small groups are some of the most abundant. So the blues that I've highlighted here are all of those picoplankton, less than 2 microns, that we can't currently measure very well, and that are the most abundant in most of the ocean. This is the North Atlantic through the South Atlantic, cutting the Atlantic. We've also tried pushing the larger size end of the size spectrum. This is a diatom that we collected on a recent cruise in the Southern Ocean, and this is a chain of diatoms that we collected on that same cruise. So these are now 150 to 200 microns, and in order to get these scans where you can see we're still not seeing a good iron signal because we still have low iron concentration. So we need a couple second dwell time. But these scans took a couple hours for each cell. And that means we can't get through very many cells in a beam time allotment. And that limits both the communities that we can measure and the statistical power of measurements. So that's another one of our hopes for the upgrade, is that we'll be able to measure more cells because of the faster beam and with higher intensity using shorter dwell times. Even within the small size range that we can currently measure quite well, this stack of 5 Thalassiocyra cells are in that sweet spot of about 10 to 15 microns. But there's a lot of variability cell to cell in the iron signal. And so the more that we can start to, the more cells that we can analyze, the more we can start to understand what accounts for that variability within groups, as well as covering more groups that we currently can't measure. On the right here, I have a scan, a large area scan that I did last fall, where I was looking at those Osteococcus, those 1 to 2 micron cells. And I just did a large scan to test this idea that we could someday, maybe, with a stronger beam, scan our full silicon nitride window. And then instead of going through with our visual microscopy initially and identifying the cells that fluoresce with chlorophyll, we could get the elemental contents of everything that we've centrifuged out of the water column. And we can start to understand better how the visual microscopy matches up with the elemental content. Do dead cells lose their iron pretty quickly? Do they retain iron for a long time, even after they're done fluorescing? We don't really analyze, look at those sorts of questions. I'm getting close to the end, so aware of the time. Yeah, like many of you yesterday, I think the higher throughput is very appealing, but for us, it's not just for statistics, it's also to be able to analyze this, cover more of the community that is out there. So that is all I have. I think there is time for a question or two. If there are any.
Yes, yes, we have a couple minutes for a question. Great talk, by the way.
Thanks.
And do we have any question?
Yeah, see?
Go ahead. Yeah, go ahead.
Yeah, hi Larry, great presentation. I wonder about, because you are using the synchrotron to measure the metal concentrations from the individual diatoms, let's say. I wonder about whether you have a chance to do a comparison between the synchrotron measurement and other techniques, something like ICP-MS for population measurement, and then whether there's any results or conclusion or anything.
Yeah, that's a great question. So we can do ICP-MS analyses of a bulk population from a culture. So we can match up synchrotron. That was actually that this scan of Osteococcus was comparing, this was a culture that I grew. And so I was comparing the Osteococcus here with an ICP-MS measurement. But when we're looking at our open ocean samples, those grids contain so much diversity that, that if we took all of this and digested it, we don't have a way to only measure cells. So what we can do is we can measure everything that's chemically labile to a weak acid leach, acetic acid hydroxylamine leach. And that gives us not only cellular material, but also some we call it authogenic iron, minerals that precipitate in situ, but that are also fairly chemically labile. And the cellular fraction of that is only about 10%. So yeah,
Mhm. Yeah, that makes sense. Thank you.
thanks for the question.
Thank you, Laura. And we're going to move on to the next speaker.
Yeah. Can I share my screen?
And yes, please go ahead. So our next speaker is Dr. Mary Izzo, is an associate professor for the IPIM Institute at Paul University in France. She received her PhD in geochemistry from Grenoble University in 2001. Her research is focused on environmental science, and particularly the speciation of metal in soils, sediment, and biota. She develops a multi-scale approach going from the field and the culture analysis to the microscopic and atomic level, with a particular interest in chemical imaging and X-ray absorption spectroscopy. She was granted with a 6-month Fulbright fellowship to conduct research at the bio-nanoprobe beamline and advanced photon source at Argonne in 2022 that worked with Su Chen. Go ahead, Mary.
Thank you for the presentation, Lou. So can you hear me well?
Yes.
Yeah. Okay.
Yes.
Okay, good. So my talk is about the nanoimaging to tackle microwave transformations and methylation by bacteria in the environment. So this work was done in collaboration with students and collaborators in my university in Pau in France, and together with people from APS, of course, and some people from ESRF in France. So the context is related to the contamination of mercury, the contamination of the environmental with mercury, which is a global pollutant because it transfers through a large scale in the atmosphere. And mercury is one of the most concerning contaminants on Earth because it transforms into methylmercury, which is a strong neurotoxin. So the methylmercury is produced by anaerobic microorganisms in aquatic system and in anoxic sediments. And methylmercury is mainly produced by sulfate-reducing bacteria, SRB. So the produced methylmercury is then bioamplified and bioaccumulated in the aquatic food web. And so there is a risk for the consumer at the top of the food web. So what we know now is that 2 genes are required for mercury methylation in bacteria, AGCA and AGCB. But however, it was found that mercury methylation rates were not correlated to the expression of these 2 genes, and also depends on environmental parameters such as the mercury concentration and sulfur concentration. So the mechanisms of mercury methylation are still poorly understood, actually. So our global objective is to progress in the understanding of the mercury methylation by sulfate-reducing bacteria. And for that, we study an original model strain, SLB model strain, Pseudodisfibrio hydrogeri, called Baroque 1 strain that was isolated from the Bear Lagoon in France. So it adds a new model in the study of the mercury methylation. And so we have specific questions. So what are the mercury species involved in mercury methylation? And what is the mercury distribution at the cell level? It can give information about the mercury methylation pathway. So it was shown in other strains that mercury is methylated inside the bacteria and is then exported out of the cell. We don't know exactly the role of the surface ligand, and we don't know if mercury is accumulated in the bacteria cell. And as bacteria have 1 to 2 micron size, we need nanobeams to focus on this cell. So we studied 2 bacteria strains, our model, Baroque 1, that methylates mercury, and another strain, SRB strain, G200, that doesn't methylate mercury. So we work in anaerobic condition, in non-sulfidogenic condition, actually with fumarate and pyruvate medium to limit the precipitation of mercury sulfide. We follow the bacterial growth by optical density, and we expose the bacteria to mercury at the beginning of the exponential phase. So we study a range of mercury from 50 nanomolar range, nanomolar to 50 micromolar. At the end of the incubation, so we measure the production of methyl mercury and inorganic mercury by gas chromatography ICP-MS after isotopic dilution. We then studied the partitioning of mercury between the pellet and supernatant after centrifugation, and again measured the concentration of methyl mercury and inorganic mercury in each. And we collect the cell pellets to identify the mercury species in the pellet. So we do that by cryo high energy resolution fluorescence detect scanners, as Sue mentioned previously. And we studied the mercury localization by electron microprobe and also cryo X-ray fluorescence microscopy. So this is the effect of the mercury concentration on the methylation. And so this is a graph. So the concentration here is expressed in micromolar per cell. And this is the production of methyl mercury. So we see that the production of methyl mercury increased in Baroque 1 until a saturation point, which corresponds to 2.5 micromolar in the medium culture. And after that, we did not measure any production of methyl mercury. And for the highest concentration investigated, about 50 micromolar, the growth of cell was strongly affected since we could measure only 15% of growth. So there is a saturation of the mercury methylation process with the concentration. So we then move to the partitioning. So in hot red, this is the methyl mercury in the supernatant, and in hash black, this is the inorganic mercury in the supernatant, and in plain gray, this is methyl mercury in the pellet, and in black, the inorganic mercury in the pellet. So for Baroque 1 at the left, so we see that for the lowest mercury exposure, we could measure methyl mercury, and that was found in the supernatant. So that means that the methyl mercury is exported after the cell. And for the high concentration, we did not measure methyl mercury, so in agreement with the inhibition for higher exposure. So when we look at the inorganic mercury, we see that for the high exposure, it was mainly found in the cell pellet in contrast to the lower exposure. And when we look at the non-methyl strain, G2O, of course, we did not measure any methyl mercury in it. And when we look at the partitioning, we see that the main part of we see that the inorganic mercury was found mainly in the cell pellet. So one question was, what was the mercury species involved in these pathways? And so for that, we use high energy resolution fluorescence detecting xanes. So we did that in Grenoble, and just a few words about these techniques. So in conventional xanes, actually the fluorescence is detected directly from the sample, and we measure so both the L-alpha-1 and L-alpha-2 lines for mercury, as well as diffuse signals. And with high resolution, there are some mirrors that are set in drag position, and only select the mercury L-alpha-1 line that is sent to the detector. So this is in the picture, you see all the mirrors that are used at USRF on FAME beamline. So here the graph shows a comparison of conventional xanes in blue and in black for high resolution xanes, and we can see immediately the differences between both techniques. So we see that many spectral features show up for the high resolution in black, and this is really important to make the difference between species with sulfur because sulfur is strongly involved in mercury speciation. So we synthesize many mercury compounds, methyl mercury and inorganic compounds, and we collected the high resolution same signal for all these compounds. So we see, for instance, that we have a peak for methyl mercury species that are bound with sulfur. The peak is located at the different energy that mercury bound to sulfur in diagonal position, for instance. And we see a particular age for the mercury tetracoordinated with sulfide. So it shows the good, the high precision of the techniques. So we collect the signal from barrow points, so the methylene strain exposed to various concentrations of mercury, and then to the non-methyl strains in red here. And so we use linear combination fitting to find the species. And so we found that for low mercury exposure at the bottom of the graph here, we could measure methylated mercury with cysteine in red, and that the main part of mercury is found as tetracoordinated mercury sulfides, that is beta AGS or metacinnabar. When we increase the concentration of mercury, we see that the speciation changed. There is no production of methyl mercury, but we see that for non-growing cells, the mercury species drastically changed, and we identify mercury 0 in this culture. For the non-methyl strain, the metacinnabar is still the dominant form, but we also identify some tetracoordinated mercury with cysteine. And again, for the non-growing cells, we observe a drastic change with the occurrence of mercury 0 in this case. So we observe this beta metacinnabar, and one question was is it present at the cell level or not? And so for that, we use cryo X-ray fluorescence microscopy on bio-nanoprobe beamline. So working with C-chance. So for that, bacterial cells were deposited in silicon window, fast frozen in liquid ethane, transferred with the cryo transfer system, and then analyzed with liquid nitrogen at the liquid nitrogen temperature. So the resolution is 80 nanometers at BNP, and we work at 12.3, give a step of 65 nanometers with quantum rate of 100 to 200 milliseconds. And for our last experiment, we also tested the tychography. We'll talk later about it. So we expose the bacteria to 2 micromolar of mercury during 4 hours, and we did not see any impact on the cell growth. So that means that it is not so much toxic at this concentration. So we can observe several cells here. Well, the potassium is quite visible on this cell. We see also sulfur and also some iron, and particularly some spots of iron. I will come back to this iron later. And when we look at mercury, we just see some extracellular spots co-located with sulfur, but not present in the cell. So this is another picture that we collected. Again, we see several cells with potassium and sulfur. And when we look at mercury, so we see, well, in the tricolor maps here, we see that mercury is present as extracellular spots with sulfur, potentially attached to the cell membrane. And we see that 2 cells are very rich in mercury. So we still see this extracellular beta AGS, and we suspected that these cells, very enriched with mercury, were dead. So to check that, we decided to use a multimodal approach in the beamline and to combine, so in the beamline, cryo-optical fluorescence and X-ray fluorescence microscopy, as well as spectrography. So for that, we use 2 dyes, the cytonine that enters the cells and which that emits in green, and the propidom-iodine that only enters the dead cell because the membrane is damaged, and that emits in red. So this is a picture that we collected directly on the beamline. So this is the picture at the left for the green probe. So the live cells. And so we can correlate the image obtained with optical fluorescence to the map collected with XFN. And we see in these three cells, we see potassium here, as well, sulfur. And again, we could not see mercury in this cell, in this live cell. So we could observe only extracellular spots, nanospots, we collocated again with sulfur. So to focus on this live cell death, so in our lab for our last experiment that was 3 weeks ago, just before the shutdown actually, we pre-selected some cell with a cryo-optical fluorescence in our lab. And so in green we have the live cell, and in red we have the dead cell. And we took the coordinate of the cells to use the same samples and the silicon windows on the beamline. So this was really important to pre-selected our sample. So unfortunately, we had an issue with the cryo system on the beamline just before the shutdown. And so we had to freeze dry our samples and to work at ambient temperature. However, it was not so bad because we can find the same cell in the windows even after freeze drying. So we can locate the green cell. So it was not so bad. And then so we took the map of these cells, a red cell and green cell live, and we see that potassium is present in these three live cells and was not present in the dead cell. And when we look at mercury, we see that the mercury is present in the red cell, so in the dead cell, and not elsewhere. So we took the typography on of these three cells, and we see that the two cells that were supposed to be live have a good structure, and in contrast to the red one, which seems that is damaged. So to understand more these nano extracellular spots of mercury with sulfur, we moved to electron microscopy and microprobe with Nestor Saluzek at Argonne. So this is an image of 3 bacteria in dark field. So we see that these bacteria have a very bright spot inside. So this is not mercury, actually. This is the iron with phosphorus that are present inside the bacteria. And when we look at the extracellular medium and with the membrane, we see very small spots here. This is nanometer spots with 2 or 5 nanometers. And these spots in red is really enriched with mercury. So these nanoparticles sometimes form aggregates here for 10 to 20 nanometers with mercury. And if we look deeper, we see that this material is crystallized with, and we can measure the lattice parameters which match, which is 3.3 angstrom, which match with the beta IGS. So to conclude, mercury is present as mercury sulfide nanoparticles at the extracellular, as metacinnabar. Most of the cells do not accumulate mercury, and so the mercury produced is exported fast, and maybe inorganic mercury is exported too. And some cells accumulate mercury, but are likely dead. So what we expect for the APS upgrade is an increase in resolution. So it is interesting to probe the intracellular compartment. In our case, so the membrane periplasm, the nanoparticles, and the organelles. With a higher flux, we can expect to detect dilute elements. This is really important in environment where toxic are usually present as trace elements. So we can work with more relevant environmental conditions. Maybe we can have potential issue from beam damage, of course, because the dose will increase. So it should be important to work with cryogenic capabilities, to use speeding freeze, and try to monitor the samples. So tychography can be a way to do that. We expect also a correlative imaging with a cryo setup. And I would like to mention also the importance of cryotransfer system that can be used for our cryo samples pre-characterized in our labs. So finally, I would like to thank everybody, especially at Argonne during my visit last year, and the fundings. And last slide, I would like to do a little bit of advertising. So we see Chan at APS and Jean-François Gaillard from Northwestern University. So we created a research topic on advanced techniques for the chemical imaging of metals in environmental science. So from what I heard today and yesterday, I see that many words can match with this research topic. So please go ahead. Contributions are welcome, and the paper are expected for September 23, and they were published in Frontiers in Environmental Chemistry. So feel free to contact us if you have questions about that. And thank you for your attention.
Thank you, Mary, for the great talk. Do you have any question? Okay, I have a very simple question for the Herfty measurement. And how many those fancy mirror you had in the setup?
12.
20. Okay, what kind of detection limit can you push for this kind of setup?
So it was measured for selenium. It was around, it was less than 1 ppm. So for mercury, it was not measured precisely, but for selenium, it was just less than 1 ppm.
Thank you. Do you have any question for Dr. Izzo? If not, then we can take a break, and that's close the morning session, and we'll come back at 10:40. Our first speaker for this second part is Dr. Swarpicina. And Dr. China got his PhD in atmospherical science from Michigan Technological University. And he leads the Territorial Atmospherical Process Integrated Research Platform at the Pacific Northwestern National Lab. His significant research area include atmospherical aerosol property and processes, and atmospherical aerosol chemistry, and heterogeneous in ice nucleation and aerosol cloud interactions. Today, his talk is going to be the chemical imaging of atmospherical particles. Please take over, Dr. Mark.
Thank you, Zoe, for the introduction and for the opportunity to share my work here. So as you mentioned, my background is in atmospheric science, so it's a little bit different topic that you guys have been discussing since this morning. So I'll talk about how the chemical imaging help us to understand several atmospheric processes, and I'll try to give some background information so that will help to understand some of the slides that I'm going to go through. So the main research area that we study is called the terrestrial atmospheric processes, and I just want to briefly show the schematic where kind of get an overall picture of our work here. So when plants and soils or any other terrestrial sources emits particles or gases, for example, water organic compounds into the atmosphere, and then there's several type of rapid processing happening in the boundary area from after reaching after the emission and before reaching to the cloud, where these particles help to make a cloud droplet. So particles can be either both primary emitted, for example, different biological particles from the plants and soils, or it can have a different secondary organic aerosol, meaning that when you have different water organic compounds emits either from plants and soils, they can get oxidized in the atmosphere and create the secondary organic aerosol formation. And what we really want to study what is happening in this layer and before reaching to the cloud, and how this particle affect the cloud formation, and specifically the ice formation, which is very a very uncertain phenomena that we have in the atmospheric science community. And why we care about this, because 50% of the Earth precipitation comes from the ice phase. So you can see there's a big impact on the hydrological cycles to try to understand these processes. So the four areas that we study under this terrestrial atmospheric process are land atmosphere feedbacks. So meaning that when you have emission from the lands and then you get into the atmosphere, and then atmosphere again try to get deposit those constituent back to the surface. And there are different gas exchange processes are happening. And biosphere atmosphere interaction is very similar, but here we're talking about specifically the biological component like plants. And then molecular understanding of aerosols, try to understand what are the different molecular mechanisms that that we can explain, let's say, to define processes. And then finally the aerosol cloud interaction. So for this talk, I'll mostly focus on the biosphere atmosphere interactions part. And to give a little bit more background of the atmospheric aerosols, these are highly complex. These are multi-component, so you don't find a single component where you see just one compound. So they are always mixed in the atmosphere. That's why it's make it really challenging to study those, and that's the reason we need a multimodal approach. So for our group, we use varieties of microscopy, spectroscopy, and high resolution mass spec to study these atmospheric particles. So one of the things that I'm gonna focus a little bit more today about this biosphere atmosphere interaction component, what I mentioned is this spores or pollen particles, like as we all know, during the allergy season, especially now the spring season, we have a lot of pollen in the atmosphere, and then it gives you the allergy. One of the things that we try to study, and something that was not, getting not much attention before, especially in the atmospheric science, because these particles are quite large, like 20-30 micron. So their residence time in the atmosphere is quite large. So there's not much atmospheric relevance for those large biological particles. But what happens is that when these particles get into the cloud, they ruptured and they get into very small tiny particles, and then these particles, the size range, and the atmospheric science community are really interested, the submicron size range, where these particles can promote cloud formation either in the warm cloud or in the cold cloud. So one of the things I wanted to show you today is this rupture event of the biological particles. So not necessarily you need to get into the cloud, but let's say if your particles are exposed to high relative humidity conditions, these particles get fragmented and release these small tiny particles, and they have very wide range of composition. And I'll show you some of the images that make it really challenging to detect those particles once they emit into the atmosphere, because some looks like just a salt particles that you expect from under the marine environment, for example. So this make it very challenging to detect those and then understand their impact on the climate. So one of the studies what we did is that we have a fungal spores. Those are actually collected in one of the DOE project where we sampled in Amazonia rainforest, and then we exposed them into high humid conditions, and then you see this. You started to see these different ruptured particles, and then we try to probe those their size distribution. And if you look at x-axis is the size, and y-axis is the frequency, and you can see this is the blue one is the size of the intact fungal spores, and when you expose them into high relative humidity, they tend to rupture, get into small pieces, and you can see they are below, sometimes below 100 nanometer to up to 1 micron. And again, this is just a rough estimate. We need a better understanding or better measurement of the size itself, but this is kind of to give you the first picture of what to expect of those fragments. What was one of the interesting part was puzzling for us is that these particles, when you observed, we see a lot of sodium in those, and our expectation was these are coming from salt. But as we can see from the images, these are indeed fungal spore particles. So that's why, come into the multimodal imaging part that, we just, for example, scanning electron microscopy with energy dispersive X-ray spectroscopy, you see, peak of sodium, because sometimes they are very close to the copper, which is coming from our substrate, and make it very difficult to confidently say these are indeed sodium or not. So then we use other soft X-ray technique or our secondary ion mass spectrometry technique to detect those particles, and we found that high abundance of sodium rich particles, which kind of a new avenue that gave us that these are not necessarily the marine environment, but these are indeed coming from the enriched biologically region. And when you look at the fragments, and these are looking at the sodium map of the fragments, and you can see this very highly localized sodium constituents. So these are indeed showing the presence of sodium even when the fragments in the atmosphere. So then I'll move back a little bit to the pollen one that we started. So this is kind of our ongoing study, and then I'll mention how the APS has been helping us on this aspect. So when we look at the spores, so spores are typically difficult to rupture because you need a several round of high humid cycling, but the pollen itself is very easier, let's say easier than spores to rupture them. So if you're exposed to high humid condition, they tend to rupture into tiny pieces. And again, the same problem is that how do we detect those particles, because those are very similar to some of the other particles that you present in the atmosphere. So it's very challenging analytically kind of to detect those in the atmosphere, which we meet our understanding, and then to put a mechanism that in the model that we can, or parameterization that we can put in a model to understand their impact. So then we work with C and Zoe in the APS bio-nanopore beamline, and they did some control laboratory experiment. So what we did is in this case, we have these birch particles, but we fragment them into small pieces, and we're trying to look at their different constituents. So, and typically, pollen or any other biological constituent, the two typical indicator that we use is potassium and phosphorus. So, and typically especially the potassium and phosphorus, they have very trace amounts. So sometimes it's very hard to detect with the EDX-weighted technique. So that's why the bio-nanopore come very handy kind of to detect those trace amount, which will help us to detect those particles in the atmosphere. And you can see there's a high the contrast of the potassium constituent of this birch particles. Those are like a fragment of pollen. And then next, what we did is that we mixed the pollen fragment with our dust particles, kaolite, which is a standard dust we use several application, and because we know their constituent and structure. And then again, we try to look at those mixing, like how these tiny fragments can mix with the dust, because that's what you typically observe in the atmosphere. So we try to mimic this condition and try to see if we can detect those. And you can clearly see those again, the zoom in portion of these potassium rich particles, and sometimes you see some metals, which is kind of showing the presence of this both dust constituent as well as the biological component. So this is one example actually with the phosphorus. That's what I want to show this image here, and you can clearly see these are the same samples or similar type of experiment where we have a biological fragments with the kaolite dust particles, mix them together, and you can see the highly concentrated phosphorus, which is again very efficient to detect compared to other EDX-weighted technique. And then also you can see the presence of, for example, metals, and see how they they mix them together, and that really help us to understand there are two things. One is the detection of this biological constituent, which are very trace amounts, and second thing is that the mixing, which is very important for different atmospheric processes. Like if you have two different components, how they mix them together. And the term that we use to quantify is called mixing state. So this compositional information along with the morphological component will give us idea about the mixing state of these mixed particles. Then next, what we did is that again we did a series of this experiment, and then we tried to understand their detection. And in this case, what we did is we have the same biological fragment, but instead of dust, this time what we did is added a SRFF, which is a swine river fulvic acid, which is a standard organic that people use as a surrogate organics in the atmosphere. And here again, you can see this potassium, for example, that I can present because of this biological constituent, but there was you can see the absence of metals. So again, kind of show that there was these particles indeed you are looking into the or dominated, let's say, by the organic component, where you see the presence of potassium as an indicator for the biological constituent. So again, this is to give you an idea how different imaging technique can help us to detect this fragment, which are again still that is still an analytical challenge to understand their impact on the climate. And I just want to briefly show why we care about this specific mixing. And as I mentioned, one of our goal is to understand is how these particles, when they get into the cloud, how they make ice in the atmosphere. So here, this is an instrument that we built actually in our lab at EMSL PNNL, where you can see this is a real world atmospheric particles, and they have a mix of different constituents, and you can start seeing the nucleation event. So this each images are 3 seconds apart, and you can start seeing this on the top corner here. You start forming this little high contrast. So these are the actual ice particles. So you can see how the ice is forming within a single particle. So just to give you some idea, like why we do care about the ice formation. Again, briefly, I want to mention that once we have these particles in the atmosphere, of like some of the data that we get from APS, we can apply deep learning approaches to detect those. That we need a high number of particles, and then if we can train them, and we are able to detect with pretty high accuracy, almost 97% accuracy to detect those particles. So again, this is, I don't want to go into details, but the idea is that we need not only imaging but advanced machine learning or deep learning techniques to couple them together to get the atmosphere. I mean, to better understand their abundance in the atmosphere. I also briefly mentioned that we do also see the a little bit different ways. So we are talking about the biological fragments, but here we're talking about the dust fragment, and you have a biological intact particle. So we also observe those particles in the atmosphere, and how these particles affect the ice formation. So I just say, and I know that I'm probably running out of time, so just quickly mention that you have this fungal spores where you have 2 or 3 dust particles, and if you expose to high, colder temperature, you start forming this ice. So really kind of why we really care about this mixing of different constituents. So this is something to give an idea that these are very important for atmospheric science community. And I have like just few slides kind of to show that what are our potential future collaboration looks like with PS for multimodal imaging technique. One of the things that we are really focusing these days is a partnership with another user facility called atmospheric radiation measurement arm, where we conduct balloon launches and then try to understand the vertical profile of different atmospheric constituent. So for that, we built some instrument and to collect samples, and then we bring those back in the lab and try to study different atmospheric events. And one thing I would like to highlight quickly is the work made by our colleague Lata, where she probed those particles inside the cloud and above the cloud, and kind of show that I was expecting some images here, maybe I lost it here. So it shows the, how we can see the cloud process particles. And the last one I want to show that when you look at this a different layer of clouds using chemical imaging technique, this again soft imaging, but the thing is that we need not only just the carbon constituent, but for the higher element, we need hard X-ray kind of to detect those metallic components and some other trace amounts, so that will give us a better idea of their atmospheric constituents. So with that, I want to thank you, and sorry I was running a little bit late.
No, you're doing good. You're doing great. Thank you. Thank you very much for the wonderful talk. Do we have any question? We have about 1 minute. Quick question.
Maybe I went too fast.
That's a lot to take. Okay, and maybe if you can stay after the session, and if people have a question, and then we can ask them. Thank you very much, Mark.
Thank
and
you.
we're going to move on to our next speaker, and actually Dr. Tanya, and she just showed up. Can you upload your?
Hi, everyone.
Okay,
Do you see my slides?
hey. Yes, I'll make a brief introduction for Dr. Tanya Ponsku. Is that right? I hope I pronounced your name right.
That's good.
As a research associate professor for radiation oncology, she got her PhD from Illinois Institute of Technology. Her current research interests include radiation biology and nanotechnology and development of our new instrument for high resolution microscopy and bio-nanoprobe. Okay, please take over, Dr. Tanya.
Hi, everyone. I'm really glad to be here and talk to you. I apologize for joining in such a rush. I have been stuck in traffic for 3 hours longer than I anticipated, and I'm away on travel. So this has been quite a mess. So I apologize again. So my talk today is about the use of multiscale X-ray microscopy for evaluation of archival tissues from animals that have been exposed to radioactive particles. So what I am talking about here is an archive of tissues from dogs that have been exposed to different types of radiation. This animal tissue archive is called NURA, which is a short from Northwestern University Radiation Animal Archive. And in this archive, there are many thousands of animal tissues from thousands of dogs and rodents that were exposed to either external beam ionizing radiation or exposed to internal emitters of the type that would be released in a case of a nuclear incident or nuclear accident. This is a collection of tissues that were generated during the period between 1950s and 1990s. That is why we have thousands of tissues. This work has been done at several national laboratories, such as Argonne National Laboratory, Oak Ridge National Laboratory, Lawrence Livermore National Lab, then University of Utah, Lovelace Inhalation Research Institute, etc. And this work was sort of spurred on by concerns around during the Cold War era. However, more recently, the government has realized that there is the possibilities for exposure to internal emitters are no longer negligible, and that, while we are all hoping for peace and everything to be good, possibilities for some sort of an accident or a problem or an exposure to internal emitters are not not nil. So because of that, we have received, the lab that I work at has received funding to work on samples from animals that have been exposed to internal emitters. The reason for this focus is comes from the fact that external beam radiation exposure to X-rays or gamma rays is done both in medicine and in accident-related situations. So a lot of work has already been done in order to try to figure out effects on normal tissues when, say, cancer patient receives external beam radiation therapy. However, internal emitter exposures are something that does not happen normally. And there is very little research being done on that. And pretty much everything that was done was concluded in 1990s. So, oops, sorry. So the first step in our process of working with these samples was to select the best samples. And I will begin by showing you how this, how a specific instrument for inhalation of radioactive particles looked like. So at this Lovelace, later called ITRI Research Institute, the researchers during the 1960s had come up with this system of like double chambers where there is an opening through which an animal's nose is being put, and then radioactive material in form of radioactive particles is being dispersed in a box, and the animal can only inhale air from that box. And the animal gets a latex mask put over its head, and it was being suspended in the air so that it could not pull its nose out. And in this manner, work was done where researchers were able to measure how much the animal inhaled and so forth. And the external body contamination was presumably negligible, even though these guys later did wash the exposed animals, and sometimes there was some radioactivity in that washout. In the very early experiments that were done with these animals, what has been looked at was where the radioactive particles accumulate. And this was just done sort of by bulk measuring the amount of radioactivity in different tissues over a different period of time post-inhalation exposure. And you will see here that lung has the most radioactive particles and greatest radiation burden. However, there is also some accumulation of radioactive particles and radiation in transbronchial lymph nodes, labeled here with TBLN. And this was of great interest to us because this lymph node is one small piece of tissue that can be imaged relatively quickly versus whole lung, which is giant, and the researchers who worked with these animals had only preserved 17 samples of lung that fit within like centimeter by centimeter cube. So the possibility to get like a good overview of where the particles are and so on is much easier if you use tracheobronchial lymph nodes. And these are the lymph nodes that are in the part of lung that is close to the part where lung trachea branches into bronchi, and then the largest depositional particles, like sort of residual nanoparticles, can stay in that region during inhalation. Then the next step was to look at these samples from dogs using X-ray fluorescence microscopy and X-ray fluorescence mapping and visible light microscopy imaging, and to try to compare the two. So here is the first slide, and my talk is about a multi-scale imaging and multimodal imaging. This slide is showing you the distribution of elements silicon, phosphorus, sulfur, iron, and zinc. And then in a corner, you have an H&E image of the sample. So the areas are surrounded with black marking are the parts of the tissue where we have a high concentration of silicon. And because the particles that the dogs inhaled were basically aluminum and silicon particles that were just surface coated with radioactive yttrium-90, presence of silicon should be indicating where yttrium was located at the time when deposition of these particles happened. What attracted our attention already in this first glance screen of the sample is that, for example, the area that is in H&E image circled in red, where we have a metastatic cancer present in this lymph node, that area is not enriched for silicon. So clearly particles themselves were not present in the cancer cells, which is in the end not really surprising. On the other hand, the thing that we have noticed looking at the X-ray fluorescence maps is that there is overlap of iron and silicon, which suggests that it is very probable that these microparticles are actually present in cells that are called macrophages, because these cells are known to have a very high iron concentration. And you can sort of also see them on this H&E images as well. And H&E stands for, I should have said that, for hematoxylin and eosin staining. So after that sort of a staining, biological samples with cells are always looking purple and pink. What is maybe not so clear from this image is that there are some structures in this case that kind of look brown. So we next looked at the same sample using X-ray beam spot size of 0.5 micron. And we conducted a coarse scan where we were able to sort of figure out what was the part of the sample that we were really looking at. So I would like to draw your attention here to the fact that, for example, the phosphorus image P channel in figure A is overlapping rather nicely with the purplish signal in H&E image, which makes sense because purple color or blue color in H&E images comes from cell nuclei, and cell nuclei are the highest concentrate parts of the cell that have the highest phosphorus concentration. And so then phosphorus signal is overlapping with blue signal. If you look at the silicon and iron, you see that they are still completely overlapping. And now also when you look at the regions of interest analysis, where we have looked at a silicon enriched and silicon poor part of the sample, we saw that in addition to a different concentration of silicon, there is quite a significant difference in quantity of iron and quantity of zinc. And both iron and zinc are enriched in macrophages. On the other hand, if you look at the phosphorus signal, as you see in image in bar graph in E, phosphorus is the same whether you have normal part of the lymph node or the macrophage rich part of the lymph node. We then did again imaging with a 0.5 micron spot size, but this time instead of skipping from one spot of the raster scan to another, skipping tens of microns, this time we had scanned with 0.5 micron spot size and 1 micron step size. And so now we have an image that is rather detailed. And now when we align this image of this X-ray fluorescent map when we all look into and then look into H&E map, we see that actually silicon signal overlaps with the brown signal, with the brown image in H&E image. And that brown color is associated with so-called melanophages, which is a specific type of macrophage. So now we have this added confirmation that what we are looking at are samples that are that we probably have in these lymph nodes an enrichment for microparticles because they were brought there by macrophages. Next, we use the bionanoprobe and use the 80 nanometer beam spot size to look at the same sample. And we have been able now to detect that silicon signal is circular in most cases, and that the sizes of these areas vary between a circle which is a little bit more than 1 micron to something that is as large as 5 micron. And if you look at the particle size distribution that has been known for the sample, these microparticles go as high as 6.5 micron, but most of them are in size range between, I don't know, like 1.8 and 2.1 micron. So we have a good match. The other thing that is, however, obvious for this image now is that we now no longer have a exact overlap between silicon and iron. And that comes from the fact that macrophages are large cells, and that the vacuoles in which they deposit iron are different than the vacuoles where they will deposit microparticles. So then the question was, since we have now, we know that these are the macrophages that have taken the microparticles, the question was, did the microparticles degrade? Was perhaps this just the remnant insoluble part of the particle, and the yttrium-90 has left the particles? So we did imaging at 18.3 kiloelectron-volt energy. And at this energy, we can get a zirconium signal, and zirconium is a daughter product of yttrium-90. And we actually indeed see some amount of zirconium still present in the same places where silicon is present. And so this is a confirmation that the particle synthesis was such that yttrium-90 has remained attached to the microparticle, and which led to the fact that that the zirconium was also present there today. One thing that is maybe not very obvious from this whole discussion is the difficulty that we are having with aligning visible light microscopy images with X-ray fluorescence microscopy images. So what we have tried to do is try to come up with a process that would help us to do this alignment in a more automated way. And after we do that, we'll be able to extract the biomarker quality data from these X-ray fluorescence microscopy images and data. So here is, sorry, it is taking a while to load. Here is an image of a sample and a long and painful procedure that I use to manually align X-ray fluorescence maps with H&E maps. So I have a sample, then I have to reduce the size of the sample when I'm skipping from one beamline to another. And then I try to figure out what part of H&E image I'm looking at. And then I look into the black portion of the image in X-ray fluorescence map, because that's the part of the image where there is no signal. And I try to align shape of the black areas with shape of the white areas in H&E images. And that is not a pleasant task. It takes forever. And I am always, concerned that possibly I did not align it quite right. It is very difficult to find parts of the tissue that are very that look identical, especially when you're looking at lung tissues, which is the case in this image, because lung deforms when you are sectioning it. Here is another sample where we see these like crystals that have deposited over the microparticles. This is quite an interesting sample, but again, the H&E image and XFM images do not really overlap all that well. So we have worked together with people from APS, and we now started working with people from Northwestern University from cell imaging facility, trying to approach this problem from both ends, looking both at H&E images and visible light images on one hand, and X-ray fluorescence images on the other hand. And in conclusion, we have been sort of forced to start to look at high volumes of samples, and that is now pushing our curation of the database and forcing us to go to the new level of evaluation of our samples. And it is also pushing us to develop this rapid, semi-automated or fully automated multimodal imaging approach. And we are very excited about upgrades because that will help us to rapidly scan really large areas of the sample at submicron resolution. And the greater is the area that we have, the easier it is to actually do a decent alignment. And then the other thing that we are hoping to be able to do is to get better elemental peak discrimination and to use that to help us to figure out these like daughter products of different radionuclides and so on. So that is all that I have. I will stop sharing and I will put my camera on. I hope I do not get booted out for the questions.
Thank you very much for the talk. Do we have any quick questions? In the meantime, and Dr. Socha, could you please upload your presentation or sharing your presentation?
Yes, I'm getting ready.
Okay.
Give me 1 second.
Okay, so since it looks like there are no questions for me, I will step out. Thank you.
Thank you. While Dr. Socha tried to upload the image, I'll make a brief introduction. So our next speaker is Dr. Jake Socha, is the Samuel Herrick Professor in the Department of Biomedical Engineer and Mechanics at Virginia Tech. He earned a Bachelor of Science degree in Physics and Biology from Duke University and a PhD in Biology from the University of Chicago. After graduate school, he was the Hugo Fennell postdoctoral fellow at Argonne National Lab, studying insect flow system using synchrotron X-ray imaging and advanced photon source. His research program at Virginia Tech combined focusing on investigating the biomechanics and the functional morphology of flows in and around organisms, specifically flying snakes, frog, and insect. Prior to enter the science, he was a member of the teaching for American National Teacher Corps, working in South Louisiana. Today, his talk is a full field imaging of insects and other small animals. Please take over, Dr. Socha.
Thanks for that nice introduction. And I want to thank Olga as well for organizing this. I appreciate it. So before I get started, I would like to thank all of these people here. So over the decades that we've been doing this work, all of these people have really made things happen. So I appreciate them so much. And I hope they see this at some point. Also note the small phone here, Francesco, for those of you who know him. Okay, I want to start out here by making a point about the animals that we work on. So the majority of our work has been done on insects, and they might seem trivial to you, but they're incredibly important economically, agriculturally. They spread disease, and really they are the dominant animal on Earth. So there's millions of species, more than this million that's estimated. We're here, by the way, so we're a mammal, so we've got about 5,000 species of mammals. So really insects are quite important, and our understanding of them is kind of dwarfed by what we understand in things like mammals. So one of the fundamental questions that we address is how they do basic physiological functions like circulate their blood, like move air within their body. And the way they do that is very different than in mammals. Okay, and I'll show you some examples from these systems today. So these are some of our big scientific questions that we are addressing here. We also try to figure out how insects drink as well. So how do they take fluids from the outside and then pass it within their body? So essentially, these are all sort of fluid mechanical type questions that we're able to address using X-ray imaging. So my talk today is a brief history about how we got started in this work. And then I'm going to show you some live imaging, some technique stuff with tomography, and then I'm going to show you some examples from our work as well. Okay, so a very brief history. So at the APS, our imaging of insects was really started by Wakat Lee, who's now at Brookhaven. So in the late 90s, early 2000s, Wakat was curious about imaging an ant. He went outside, he got an ant and put it in the X-ray beam and took a picture of its head. So this is the very first image. You can see it's actually a pretty poor image, but this is a historical image. He then sent this to the Field Museum, where no one was interested except this guy, Mark Westneat. And Mark is a fish biologist, but he's also a biomechanist. And he saw this and saw the amazing potential that was there for working on small animals with X-ray imaging. So their very first paper that came out of that work was from this beetle here, where you can see its tracheal system compress, you can see bubbles in its gut moving, and this formed the basis of a science paper. I joined their group shortly after they started this work, so I'm not a co-author here, but this was on the cover of Science in 2003. And I'll talk a little bit more about this actual animal in this system later on in my talk. So most of our work, so that original work there was done at Sector 1, but the imaging program was moved to 32. So we've done a lot of our work at 32, and then we do the majority of our tomography at 2BM. So that's where we do our work. So the basis of this imaging is, I'm going to close that. Okay. The basis of this imaging is typically we're using mono beam, usually around 25 keV. There's our insect. The insect is happy because they're now contributing to science. And then we have our scintillator that's usually in the range of 0.5 meter to 1 meter away. And then there's a mirror, and then we have any kind of camera. CCD video camera, that was in the old days. We typically don't use a CCD anymore, but you can put any kind of imaging device that you want there to record the image. And so, of course, you have absorption only image if your scintillator is here, and then with the phase contrast, you get this beautiful, nice image of the ant where you can see structures, particularly the air-filled structures of the respiratory system. Those are the things that show up the nicest because of the contrast and density. So here's a typical experiment. This is kind of a crowded table here. And this one, we were heating up the insect there, and we're on a bunch of stages that allow us translation as well as rotation of the sample. This is from a more recent experiment. So this was just from this past November, I think, where the table is a little bit more clean here. Here's our camera. And you can see in this sample, it was a very simple mount where we just put the animal in a Kapton envelope and aligned it with this camera here. And then because we don't have much on our stage here, the actual motors here on this system are lighter and smaller footprint. We've also done experiments using a high-speed camera. We've done multiple experiments that way, and we sync it sometimes with a second camera so we can get an external view of what the animal is doing as well. Okay, so a very obvious question when you're doing this type of imaging on insects is what's going to happen to the insect when you put it in the beam. So first of all, we do not use the full power of the beam, so we are dialing back, because if we did, our insects die pretty instantaneously. Okay, if we really crank things up to the full power, and with the upgrade, you know, we don't need more photons per se. Okay. So let me show you this trade-off here. So this is energy here, so 15 to 35, and our sample to detector distance is from 5 centimeters up to 100 there. And you can see here that as you go down in energy, you're getting more absorption, you're getting a better image. However, at this point, we now start to have some problems with the insect itself. Okay. And then as you go down this way with your detector further away, you're also getting better contrast as well, which I'm sure you're all aware of. Typically, we're working in this 25 keV range. Okay, now why is that? Well, there's a trade-off here. So this is from an experiment where we put 4 different insect species, so fruit fly, ant, beetle, and grasshopper, and we were measuring the carbon dioxide that they emitted. And at some point after turning the beam on, they would give a respiratory signal that is a little bit different in each one, but this is essentially correlated with them dying. Okay, so we now have this time scale of the insect's death. And so what you're looking at here is as a plot of the time to that respiratory signal. So essentially the time to death here. So the bigger this number, the longer the animal is living. And this is the power density on the animal. Okay. So what you see is this very clear trend downward, that as we're putting more power on the insect, it's not living as long. And so really we would like to have the animal live as long as possible while still getting the best possible image. So the problem here though is that as you go down here in power, your image starts to get worse and worse. So usually for us, this is a question of what's the minimally acceptable amount of photons that we get the animal and still see the behavior of interest that we're trying to record and not have it interfere with the animal's natural physiology. So other things that we can do. So we use contrast agents. So this is iodine in concert with the K-edge, and I'll show you that in a second. This is simple absorption. So this was cadmium powder that we ground up and fed to the beetle in its food, and then you can now see gut movements. And I'll show you let me show you an example of that first. So this is peristalsis in the gut, where you can see those cadmium particles that are adhered to the wall, and you can now see the waves there in the gut and actually make measurements of that motion. So the K-edge imaging is with liquid food. So here we mix iodine with sugar water, and then we feed it to the animal. This happens to be a butterfly. So this is after it drank. And so if you tune your X-rays just above the iodine K-edge, then this you get this very nice contrast. So let me show you one experiment here where we're trying to figure out how mosquitoes drink. So this was actually our setup for tomography. So you can see here how small the mosquito is. This is the head of an insect pin, and the abdomen here is distended because the way we actually caught the animal was going outside of sector 32, standing outside in the dark and waiting for the mosquito to land on you, maybe drank a little bit of blood there, and then we captured it. And so here, what you're looking at, this is a tomographic rendering of the head. It's a little bit slow here, but we're turning around. We see the proboscis, that's what sticks into you and drinks the blood. And then in green there, as we fade away, those are the tracheal tubes. That's the air-filled respiratory system. And then you can see some bubbles in the gut there. And then peeling away, this is the lumen. This is the lumen of the food tube. And then you have two pumps in the head. You have a sidereal pump, pump number 1, and then pump number 2, the pharyngeal pump. And so in a cartoon version, it's here. So muscles then pull on these pumps and enable them to be independently actuated. And so so that was tomography, but we also did live imaging. So where we glue the live mosquito to a pin there, and then we put some food there and we let it drink. And what you're going to see here are results from this. And this is K-edge imaging as well. So this is regular drinking, and you can just barely see the line of food that's in there. So this is continuous drinking. This is its normal mode of drinking. But what I'm going to show you next is a mode that we discovered where what happens is you get this very large burst here where both pumps fill to their maximum, and then it's pushed back into the rest of the digestive system. And so you can see here, as this fills up, you get this very large burst. So that's what we call burst mode drinking. Why they do that, we're not quite sure. Okay, so another technique here is for visualizing fluids is to inject microbubbles or microspheres. So this was our early attempt here on a grasshopper, where we took microbubbles and we injected them into where the heart is, and the heart is just a tube along the back of the animal. And you can see here in this early image here of that there's this pulsation, and you see these big things that are whooshing by. These are actually air bubbles that were injected by mistake, but they're actually made for some nice visualization. You can see the flow there is pretty complex. Now, in our early imaging, we also saw instances of what we thought was oscillatory flow within the heart. And this was done with our CCD camera here. So our frame rate was only 30 hertz. So this was really not fast enough to resolve things. So this was a follow-up experiment many years later where we used a high-speed camera. So we have higher temporal and spatial resolution. And this is real time. And we're also injecting microspheres instead of microbubbles there. And you can see here now, those little microspheres show up very nicely. Okay, I want to show you some extremely fast imaging, and this is extremely fast for us. We know that at the beamline, they can go much, much faster, but this is a biological scale fast. And what we're interested in here is the click beetle. So if you haven't seen one before, what they do is if you flip them on their back, they have this very, very fast motion where they bend the body. So you can see it a little better in this clip. Here's the click beetle on its back. And there it now has this click, and it's enough energy to put it up into the air. So this is one of the fastest motions in biology. And in order to visualize it, we actually took the beetle and secured the bottom half of the beetle that allows this front half to move independently. And when you do that, the animal tries to escape by clicking. And so what you see is this. So this was our very first attempts back in 2005. You can see this click process here. And when you slow this down at 30 hertz, you just get a blur. So follow up a bunch of years later, we now were able to do this with a high-speed camera and achieve frame rates of 30,000 frames per second in this recent paper. We have done more recent work where we actually have gone at higher rates, but about 30 to 40,000 frames per second is what you need to resolve this very fast motion. Okay, I want to show you a little bit of tomography. You guys are all aware of this. What we typically care about is getting the most natural anatomy possible. So very often with our preps, we don't We don't inject anything to them. We don't fix them. We typically just sacrifice the animal, chill them to stabilize the tissues, and then rewarm them for imaging, and then stabilize the animal with glue or something like that. And I would like to point out that within the past 2 years, I believe it is, at 2BM, they've implemented a new system here where you can swap out the lenses on the fly. And so you put your sample in, and now you're able to start with a low resolution, large area view, and then focus down into a region of interest that you care about, without having to go back in and swap the lenses out. It is really kind of been a game changer for working at 2BM. So this is a typical slice that we get from, this is from a beetle here. You can see some muscles there. We have better images than this now, but this one is representative. But what you can see really, really nicely are those tracheal tubes here. They show up really well. They're kind of circular there. And then this is just the tracheal system of that animal. So I'm going to show you now a piece of work that is not ours. So this was done in Europe. The lead author of this paper is Simon Walker. And I'm going to show you this because it is really beautiful work, and I think kind of the state of the art in my field. And this is an insect, or this is an experiment where they put a live insect on the turntable, and then with external cameras, they were able to use the wing beat of the animal as a reference to be able to virtually stitch together multiple tomograms throughout the wing beat cycle. It is really, really phenomenal work. And so I'll show you the 3D rendering of this fly. So this is a live fly going through multiple wing beats here, and you can go through now and you can see the muscles. So there's muscles that are oriented vertically, and then the longitudinal muscles as well that you're seeing there. And this experiment yielded this fantastic result where when you cut through there, well, yeah, there it is right there. This is a tendon that is buckling every time these muscles here are pulling downward, which is pretty phenomenal. Okay, I have just a couple of minutes left, and in that time, I'm going to show you a little bit more about this phenomenon here, where you see the respiratory system collapse and then reinflate. And if you think about mammalian systems like yourself, your lungs do not collapse, your blood vessels do not collapse, and if they do, it's pathological typically, right? So we're asking here, what is this for? How does it work, and how widespread is it? These are some of the questions that we've been working on really over the past 20 years. So as a bit of background here, that science paper there did not discover that process. So this process was known to some extent from old papers. This is from 1938, where you see the tracheal system there. It is compressed, so that it's inflated, then it's compressed. But this was not widespread information in terms of understanding how how insects breathe. And the reason for that is that this work was done in the hedgehog flea, which is translucent. So the reason that this could be seen and described is because you could see through the animal, but the vast majority, 99.99% of insects are opaque. So what is going on with the tracheal system was largely unknown. And then just a little bit more background here. So this is the tracheal system of the insect, which you can see in this beautiful composition. And these tubes go everywhere within the body. They branch and branch and branch, and they reconnect. If you go down even further, so we're out of the X-ray realm, because our smallest little tubes here can be a micron in diameter, and they can get down to about 100 nanometers in diameter, which without the X-ray microscope, if we're just doing live imaging, we can't actually resolve those. And then the wall of the tracheal system itself is reinforced which is thought to prevent collapse. But as you saw from that previous video, you can actually see the collapse pretty well. And then lastly, you can see parts of the system there that open and close to the environment. Okay, so on my clock here, it has me at 20 minutes. I have lots more images that I could show, but I don't want to bleed into the next speaker's time. So if you want me to call it quits now, I'm happy to call it quits, and I'm happy to take questions as well.
Yeah, that's great talk. Thank you, Dr. Soto. And we have time for one quick question, and maybe like if we want a further discussion, we can leave it to the end of the session. So while we are asking question, can you stop sharing, and I'll ask the next speaker to share their screen. Our next speaker is Dr. Gruber.
Hi, can I ask a question?
Okay.
Absolutely.
Yes, go for it.
So it's actually beautiful. This is Andrea Jimenez, beautiful images. What is the resolution that you can achieve?
With the live imaging, it's 1 to 2 microns or so, and then with the tomography, we're reaching down. I think we're getting it's like it's 0.8 microns or so, I think is what we're at.
I see. Do you see any when the insects die? Do you see any structural damage?
I've been wanting to work on that for years. We don't actually know what's going on or why they actually die. The other really interesting question is that when we turn the beam on certain parts of the animal, they will immediately react to it. Okay, and it depends on where you hit them. I think when you hit their neurons, they're reacting more. But why is it that? What are they sensing when the X-ray beam is turned on? That's a simple question that we have no answer for, because we have not collaborated with folks who know something about that.
Thank you.
Okay. Great talk. Thank you for the discussion. And we're going to move on to our next speaker. Our next speaker is Dr. Andre Gruber. Is a professor of soil scientist and from Michigan University. He got his PhD at Moscow State University in soil science, and he is an expert in soil pore structure, soil morphology, and a multi-scale modeling water, chemical, and microbial transport in soil. So please take over, Dr. Gruber.
Well, thank you for introduction. This is a result of a long-term collaboration with Mark Rivers. For a good 10 years, we scan in his BMD13 facility, and this kind of preliminary results of our last scans, which I believe will bring more light to understanding what's going on with soil water at microscale. So this is part of our ongoing project, NSF project, microscale mechanism of controlling carbon turnover in soils. And a reason for this activity is kind of discrepancy between large scale and small scale, which we're trying to model. So we're pretty successful in modeling enzymatic activity, microbial activity in large scale, like core scale, soil horizon scale, but not much we can do at scale of pores, because it's really difficult to measure. So conceptually, we know that most microbial activity occurs in pores in the range from about 20, 30 microns up to 150 microns. And there are several reasons for that. So one of the reasons that in these pores, we have kind of optimal contents of water content and oxygen. So it's ideal reactor for most biogeochemical processes. So smaller pores are not accessible for microbial community. Only some individual bacteria can get into these pores, or they can inject enzymes in these pores, but they cannot use benefits of this pore space. And in larger pores, we have predators which grazing, and there is very little chance for microbes to survive in these pores. So in this publication, we're trying somehow to link processes at pore scale with kind of a general idea about water content distribution. So that's why we have EF values here, which are a logarithm of matrix pressure, which can be related somehow to pore size distribution. So we still kind of using Selenium-like capillary structure, which Andrei has shown us yesterday in his excellent presentation. So this kind of capillary system, which we believe exists in soil, with only difference that pore porosity is much less than in xylem system, so pores occupy less than 50% of total volume. But this is really a good illustration of what's going on. So basically, large pores occupied by predators. There is not much bacteria living in these pores, but this is a conduit of fast water and nutrient fluxes. So these small pores are not really available for microbes, so they living in kind of intermediate pores and enjoying fluxes which coming from large pores, and carbon which can be released and decomposed by these microbes goes into finer pore and not accessible for decomposition. So basically, water and nutrient fluxes are controlled by two functions. We call them hydraulic functions. So on the left, it's soil water retention function. It's a relationship between soil water content and matrix pressure. And second one is unsaturated hydraulic conductivity. So if you will look at kind of classical Young-Laplace equation, so higher matrix pressure, more saturation of fine pores we have. And then by adding water, we saturate larger and larger pores until full saturation. So drainage goes reversely. So first we drain large pores, and then we drain intermediate and small pores. And this is kind of Laplace equation, which establishes a relation between size of pores and capillary pressure in these pores. So unsaturated hydraulic connectivity is a really important function because it's control rates of fluxes. And it depends on saturation of soil pores. So more pore saturation, we have larger fluxes we have. And second, also connectivity of pores, which is not really counted in this model, but somehow counted by soil water retention model. So there is at least two contradictions to kind of microbial habitat volumes and this water retention model. And first one is that pore sizes which corresponded to PF 1.8, which is considered kind of optimal conditions for microbial habitat, are not necessarily correspond to optimal condition of air and water. For example, in sandy soils, we have much less water content than in clay soils. So it's optimum, not so we cannot directly translate large size of pores to water saturation. And second, this model doesn't explain high diversity of microbial community within pore space. So from publication, we know that about 80%, from 80 to 90% of visual porosity is connected. So where this diversity comes from, why they do not communicate, so why don't they share resources? So Okay. So our hypothesis was that microbial interaction, so pores, is controlled by pore water connectivity rather than pore space connectivity. And that's why high diversity of microbial communities comes from. So it comes from fragmentation of water, not from pore network. So our objectives were first to develop approach to quantify pore saturation by liquids and access special characteristics of soil pore network and also pore water distribution in this scale. There are two general methods to quantify soil pore water. So one is neutron radiography. So this is illustration from Ahmed 2018 publication. But this method is very time consuming and also need to be combined with X-ray CTs to visualize pores and quantify where liquid goes. So another method is dual energy monochromatic synchrotron X-ray micro-CT using dopants. And this is from publication one Windows 2002. So this method doesn't require additional scanning, but it's still semi-quantitative and mostly used for liquids, to separate liquids from gases and solid phase in pores. So unfortunately, these both methods work well for coarse textural materials like sand and doesn't really work well for clay soils. So we tried both methods. So first we tried neutron radiography and PSI Switzerland. So we incorporated leaf discs, it was soybean discs between soil aggregates. So we scanned first dry using X-ray CT, and then we scan using neutron radiography. We've got some kind of idea about distribution of water in these images, but unfortunately we were not able to quantify amount of water because of really poor resolution of this picture. Much better results has been obtained in Argonne using iodine as dopant. So we added dopant in column with soil fractions from 0.5 to 0.1 millimeters. And we placed a leaf disc on the bottom of this system and bring the system to 3 different levels of soil water content. So then we calculated, using dual energy approach, we calculated saturation of voxels in these images, and that's what this scale shows. So we see much more saturation of leaves, even in dry soil, than in soil itself. So it's allowed us to develop some kind of sponge effect of plant residue on decomposition of organic material in soil. So how we do that? So we just use a traditional approach. So we scan sample above and below KH, and then use subtraction. So theoretically, subtraction should give us an image of only iodine because it has a high increase in mass attenuation coefficient of this energy. And this is an example when iodine was placed into pipette tips and placed into the soil. So it's 2.5% of iodine solution of 5 and 10%. So when we look at the histogram of distribution, grayscale value distribution, these histograms were really broad and not suitable for calculations. So much better results has been obtained when we use median filter and then Gaussian filter, which narrow down this histogram. And then we try to correlate peaks of grayscale value with theoretical values. So we found really good linear relationship, and standard deviation of which characterize broadness of this peak decreased with using filters. But what was interesting here, so slope wasn't 1, and we had some kind of intercept. So we still don't understand why it doesn't really deviates from theoretical value. So in our calculation, we used further calibrated values of relationship between saturation, percentage of iodine, and G value. So then we developed a mass balance approach. And the reason for that, so if you look at histogram of above minus below image, so it has kind of symmetrical shape with kind of increase here, which is our signal, but it's really difficult to say where noise, a signal goes to the noise. So we need kind of threshold G star value below which one saturation of iodine would be 0. And one way to do that is to calculate cumulative value of delta G, starting from highest values and goes to negative values. And so on the left side, this is a cumulative value. But on the right side is a value which corresponds to dopant, which has been added to the whole system. So we see is concentration, this is a saturation of dopant, mu is mass attenuation coefficient, and R and F is resolution scaling factors. So when this equation get true, correspond to G star value. So below G star, all saturation of voxels is 0, and above equal to G value get measured from image divided by calibrated G value. So it's pretty simple calculation which can be done in image Fiji and really fast. So this approach has been tested and works pretty well for standards and for real soil. So that's map of saturation, which was derived from above minus below image. So now let's go to real soil. So in this study, we used soil from Maryland. It's sandy loam soils, pretty, had a lot of cracks and biopores. So we used iodine at different water contents. And dark gray corresponds to iodine. It's above KH energy. Well, no, it must be below. And so what we see here, what is really important, so we see that large pores are saturated earlier than fine pores. And this was kind of surprise for me because Darcy theory cannot describe this kind of saturation. So we use these images to calculate pore characteristics and also characteristics of distribution of liquids in this pore space. Yeah, I forgot to mention that we use 10% of iodine solution for 2 reasons. So first of all, it's pretty well visible and really good for calculations. And second, concentrations above 10% destroy pore structure. So it's make kind of sequential imaging impossible. Okay, that's a 3D picture of the same sample. So red color denotes pore space, so, and blue is our iodine solution. So we see that we first saturate small pores, and water iodine goes to cracks. But what is interesting, biopores remain empty, even at very high, almost close to total saturation of this pore system. So now we would like to characterize pore space and also liquid space. So unfortunately, in soil science, we don't have definition of single pore. So we don't know where pore starts, where it ends. And one way to calculate pore size distribution is using inscribed stairs. and calculate size of these spheres, and then probability or distribution of these sizes would characterize pore size distribution. So this kind of slides which show distribution of pore sizes in micrometers. So it goes from 0 to 87 here. So if we assume that, this is pore size distribution, which we derived from our images, and relative pore volume decreased with fraction of relative pore volume decreased with increasing size of pores. So that's normal. That's what we see in most soils. So now if we assume that capillary model works, so we should have kind of a frontal distribution of water content. So water goes first to very fine pores, here blue, then it goes to magenta, these pores, then red, and then finally goes to yellow, which correspond to large pores. But in reality, when we calculated saturation of these pores, we got this kind of distribution. So in many instances, large pores had higher saturation than smaller pores. And they come saturated much earlier than small pores were saturated by water. And this is not just in one core, so this is kind of replications of this. So in two sub-samples, we've seen pretty the same trend, but we haven't seen it in core 2. And only reason for that this core has a much less visible porosity than these two cores. So basically all dopant entered small pores. That's why we don't have saturation of large pores here. So now let's look at these characteristics. So first we calculated gamma function, which is connectivity of cell pores here. So n is total number of clusters, water clusters or pore clusters, whatever. So n is number of voxels in each cluster, and np is total number of voxels in the whole system. So what we see here is that connectivity of cell pore water increases almost linearly with saturation of visible pores. So we're talking about pores larger than 10 microns. So I would expect kind of sigmoidal shape of this pore. So when we have really low connectivity at low water content, so we have a lot of clusters, but low connectivity, then fast increase, and then kind of plateau here and high saturation above 0.7 or 0.8. Well, unfortunately, we don't have enough data to prove that, so far we have these results. So second really interesting is distribution of a number of disconnected water clusters. So this number increases initially with increasing water content, and after that decreases. So it's kind of bell-shaped relationship. And this relationship can be explained by this kind of simulation published by Führer and Bartolozoglu. In 2018, they used lattice-Boltzmann model, and they show that water is not going to find pores. So it creates kind of cluster system. So these are 3 different levels of water content. And that's pretty much what we've seen here. So basically, first we have disconnected clusters, and with increasing number of clusters, this water creates kind of water body connected system, and finally all pores are connected by water at full saturation. So that's basically explain shape, bell-shape relationship which we observed in our study. And the last result is a relationship between a number of disconnected visible pores and disconnected water clusters. So first of all, so this relationship has negative intercept, and it's understandable. So when pores, visible pores are empty, we still have a lot of disconnected pores, not saturated. But slope was 1.1, it's more than 1. So that means that with increasing water content, we have faster increase of clusters. So that means some pores in our system getting several clusters, disconnected clusters of water. So one reason for that is that we observed decrease of soil clusters, pore clusters with increasing water content. So in clay soils, because of swelling, part of visible pores goes to their size decreasing and they become invisible. So that's why technically with increasing water content, we have decrease of number of porosity and decrease of number of visible pores. So that's partially explains this slope. So then what should we do with these results? So now we know that Darcy approach, which has been used commonly for large scale modeling, doesn't really work for our scale. So probably we should go with Lattice-Boltzmann model of water distribution and also microbial interaction and modeling carbon transformation and fate in pore space. And there's some advantages and disadvantages of this model. So this conceptually better approach for this scale and big area for applying percolation theory and potentially greater flexibility for modeling spatial and temporal microbial interactions like development of hotspots of microbial activity. But it's still difficult to do measurements at this scale, even with development of demography approach. We can map enzymes at resolution of activity, enzyme activity at resolution of 15 microns, but it's not 3D, it's 2D approach. There's also
Andrew, Andrew, can you please wrap up?
I'm coming to conclusions, right? And this the disadvantage is this computationally expensive model, numerically unstable, a lot of water content, and difficult to for upscaling. Yeah, so we learned a lot, which I mentioned already in from this study. And do we really expect anything from beamlight upgrade? Well, our dream is larger volumes, finer spatial resolutions, and faster scanning time. But unfortunately, with new updates, we don't have any expectation for BMD13 line. Probably we should collaborate with others. Yeah, that's just acknowledgement. Thank you so much.
Okay, thank you for the fantastic talk. I would like to, since we're running over time, so I'm gonna like to introduce the last speaker for this workshop. And then for all the speakers, if you could stay after, and then if we can open the discussion for everybody. So our last speaker and Fabricio, could you please upload your talk?
Okay, give me 1 second.
Okay. While you're doing that, I'll do the introduction. So Mr. Fabricio Marin is graduated from DePaul University and Illinois Technology in 2018 with a bachelor degree in physics and electrical engineering. He began his career at Argonne as a scientific associate where he developed APS control tools, fine motion controls for microscopy scanning stage, and developed imaging process software. His interest in electronic and instrumentation extended outside the laboratory, where he pursued a hobby in 3D design, 3D printing, and computational photography and automation. Okay, please take over, Fabricio.
Okay, thank you for the introduction. So for this presentation, I'll go over the workflow that we have set in place at 2IDE to process X-ray fluorescence tomography datasets using Python software that we developed. And now this software is based off of a previous work called Maps to Tomopai by Young-Pyo Hung. Okay. At 280E, we use a vortex fluorescence detector to collect spectra information on a per pixel basis with resolutions ranging from like 200 nanometers to a couple microns. In the example here, we looked at battery particles, and the benefit of using fluorescence coupled with tomography is that it allows us to map elemental distributions in 3D space, and then we can also see structural information for multiple elements. The drawback, however, is that each scan can take several minutes to complete, as opposed to a few seconds or milliseconds with full field tomography. The other challenge is sample position errors. Using a similar set of battery particles as an example, the image on the top left shows an unaligned set of projections, and the ones on the bottom are aligned. The plot to the right shows the horizontal displacement errors in orange, and the vertical position errors in blue. Our scanning stages are all encoded, but however, the controller will not be able to correct for disturbances or errors that are not on the same axis as the encoder. So we kind of need some way to compensate for these position errors. Otherwise, we won't be able to correctly reconstruct a 3D volume from the set of 2D projections, which brings me to the solution. The software we developed is called XRF-TOMO, and this pulls together several image registration methods to align our datasets, and it uses Daniel Ching's Tomopai software to run tomographic reconstructions. It also validates alignment quality at the end using a number of different metrics. All of this is done interactively with the help of a GUI, and is compatible with Windows, Linux, and Mac. However, with Mac, there seems to be a few bugs, which I'm still working on. So when you first launch the software, you'll have to open up to the directory where you store your data. Now, this software was originally developed with our file format and structure in mind. So before it used to only accept H5 files, but now you can go ahead and upload TIFF images. Now each file corresponds to a single 2D scan, which we call projections, and each projection has an associated angle value. If you load TIFF images, then you'll have to also load in a text file that contains the angle information for each projection. And then once you select your data, hold on, let me see if my pointer is showing. Yeah, so once you select your data, you can choose which elements to load on the right-hand column. And then once those files are loaded, you can proceed to the next tab where the data is prepared for alignment and reconstruction. Some of the alignment methods rely on the assumption that the background values are dark and that the features are bright. And if they're not, then you'll need to click on the invert buttons to do that. And that's this button right here. And then cropping, or some of the alignment cropping is optional. And lets you reduce your field of view for things like local tomography, or if you scanned too wide of a field of view. If you find that one of the images does not belong to the dataset, or it just doesn't look right, you can exclude it without deleting the actual data by using the remove image button. And then the pad edges button lets you increase your field of view by padding the image boundaries with 0 valued pixels. This is done to keep your data from moving out of the field of view or rolling over during the alignment step. And I'll show what this looks like later. And then finally, the remove hotspot button just gets rid of the pixels that have abnormally high intensity values. And once that's done, you can go ahead and go to the next tab where you can play with some of the alignment options. For each one, I can explain the pros and cons and what kind of signals each one works best with. And whenever you use an alignment tool, it adjusts your data for all the elements, not just the one that's currently in view. And so in this animation, the slider also lets you look at the sonogram for each vertical position. So starting with the cross correlates sonogram button, it's a tool that fits a single feature from a sonogram to a line. You can think of a line as a sine curve with 0 amplitude, so it's easy to characterize and visually confirm that the alignment was successful. This method works best with sonograms with single defined features, but not so good with sonograms with multiple features, like the ones we saw in the previous slide, or for features that disappear from the field of view, because then it's no longer continuous. This next tool is a vertical alignment method that takes a sum along the horizontal directions for all projections, basically to reduce a set of 2D images to a collection of 1D arrays. We then take the derivative of those arrays and then the absolute value, and what you get is illustrated in the center image. So that S shape probably comes from the rotation stage having some degree of wobble, causing the sample to move up and down over the course of the rotation range. So to fix this, imagine taking a straight edge and then pushing that straight edge along the left-hand side until it touches all of these, the edge of these bumps. So each ray gets shifted by some amount, and then you can apply those shifts to the projections to correct for the vertical position errors. This method is good for large features, but not so good with images with multiple features or for high noisy images because of the derivative operation. This next tool is similar to the previous one, except you don't take a derivative. And it works best for channels that have few isolated features, so which would give you just like 1 or 2 peaks. And then this tool calculates the center of mass for each projection as a pair of XY coordinates, and then shifts those projections until each of those coordinate pairs are at the center of the field of view. This tool works well most of the time, but it doesn't perform well for images that suffer from self-absorption. Self-absorption typically happens with dense samples that attenuate the X-ray beam as it passes through, resulting in less fluorescence photons from one side of the sample than the other. And so to the detector, this appears as if half of the, or one side of the sample has less material, which throws off this algorithm. The next one, cross correlation, aligns by shifting the adjacent projections until the difference between them is minimized. It removes jitter pretty well, as you can tell from the left-hand image to the right hand, but the local minima that it achieves doesn't necessarily give the correct alignment. So this tool is best used in combination with other ones. This tool, move to edge, is a basic yet effective one if you have an isolated particle like this one. And all it does is it moves the feature, until the top or bottom edge touches the top or bottom boundary of the field of view. In this case, I moved it to the top because the bottom side is clipped. So yeah, two of the requirements for this is that one, it's an isolated feature, and two, that the either the space above it or below it are not getting clipped. And at this point, I should probably mention that you can save your alignment progress as a text file and then load it in later if you have another tool that does image registration. You can import the alignment information from it as long as it follows the same structure as shown on the right. So basically your file names, your X coordinates, and your Y coordinates or shifts. And some of these other features I've labeled as advanced because I've had less success with some of them. But I know others who have had success, so I've included them as options for those who know how to best use them. The Find Center tool shifts projections horizontally until the rotation axis, or the R-axis, is in the center of the image of the field of view. The rotation axis is defined as a virtual vertical line where the features appear to rotate around. If you reconstruct without centering, then you get these crescent artifacts on the left-hand image, but when it should look like the image on the right. Now, the arc radius and the concavity can tell you about how much the center is off by and in what direction. So it gives you an idea of how much to shift by and in what direction. This tool takes the most prominent feature and it moves it to the center of the field of view, as you can see from these two animations. The left-hand animation is the features that are not aligned, and the one on the right is after clicking this tool. So this tool is most effective with features that don't suffer from self-absorption and that remain within the field of view. So if you're lucky enough to find a fiducial-like object like this one, then I'd recommend starting with this tool. Also, keep in mind that fluorescence data has multiple element channels, which increases your chances of finding one that meets this criteria. Even if the element is not one that you're necessarily interested in from a research point of view, it can be useful for alignment purposes. Also, if one alignment doesn't work, you can click on the Edit Undo button. It's actually not shown here. Or you can enter Control-Z to quickly undo the last action, and then try another one. So before reconstructing, you want to also define a rotation axis such that all features stay within the smallest field of view throughout the rotation. One way to do this is by shifting all your projections along a sine curve, which would result in a sinogram, or which would minimize your sinogram envelope. In the example above, I've shifted the projections along a sine curve. I'm not sure if it's easy to tell, but there's a blue curve here, and which I've constructed using a series of sliders shown here on the left. The two images on the left correspond to different elements before shifting, and the two images on the right correspond to the same two elements after shifting. And both sets of sonograms are now within the field of view. And if you look at the bottom left one, you can see that that sonogram rolls over, or it exits the field of view, which we don't want. Another way to visualize this is with the projections and reconstructions. On the top left, you can see that the projection sweeps a larger area of the field of view, and the reconstructed cross-section takes up a larger area given by the circle, even though the feature that you're interested in only takes up a small portion of the image. Your reconstruction quality is the same either way, but the one on the right takes less time to reconstruct and has a smaller file size. On the reconstruction tab, you can choose which elements to reconstruct, the algorithm to use, and the range of rows to reconstruct. You can start by reconstructing 1 or 2 rows to quickly check the quality of the reconstruction before committing a lot of time to processing the entire data set, especially if you have a large field view and a large many number of cross sections. And then near the bottom, these 2 rows here just show you the statistics, and the parameters that you would input for some of these reconstruction algorithms are given down here. I typically just leave these at the default values. And then I also tested different reconstruction algorithms with simulated data to use as a baseline to compare with real data. Here I'm comparing the information from the original data represented by the blue curve, and then I compare it to the orange data, to the orange curve, which is the simulated, which is how do I put this? It's basically the reconstructed data, which is represented by this orange curve. And pretty much I do this to validate the reconstruction performance. And in general, what you want to look for is that the orange curve tracks the blue curve, and that there's a small difference between the original projection and the simulated projection. And you can also use the mean squared error as like a guide. So if we look at real data, we can see that the mean squared error value gives us a value of 43 compared to the value of 6 on the simulated data. The scale factor refers to the ratio of the sum of the orange curve to the sum of the blue curve. And ideally, this value should be as close to 1 as possible. Again, I would suggest using the mean squared error just as a guide and focus more on the overlap, the general overlap between the 2 curves. So with that said, we're exploring other options to validate the data so we don't rely just on human judgment alone. And then another validation tool that compares the original projection shown on the left with the simulated projection shown on the right. And then you can use different comparison metrics, including the mean squared error, structural similarity, and Pearson coefficient. So to summarize the list of challenges in this workflow, one of them is identifying fiducial-like objects, fiducial-like features has proven advantageous towards image alignment, but it requires human interaction to find. Also, the self-absorption throws a wrench into many of these alignment tools, so that needs to be addressed. And then the rotation center needs to be optimized to automate, or it needs an automated function, which is not implemented yet, but I'm looking into it. And then the alignment and reconstruction validation steps needs better comparison metrics that don't rely, or that gives us more of an objective measure that doesn't rely so much on our judgment or on our validation. And then with the APS upgrade in mind, there's some things to consider with our workflow. We may want to use interferometry feedback on our positioning stages in place of encoders for more accurate position tracking. Alternatively, we can still use encoders, but also collect the interferometer position and use it for image alignment after acquisition. Other presentations have mentioned that the increased volume of data will introduce operational bottlenecks if we can't keep up with it. So one thing we should consider is to leverage existing computational resources so that we can use more powerful alignment and reconstruction algorithms and automation tools. XRF Tomo was initially built with compatibility in mind to run on the standard laptop computer, but that may no longer be practical because we need to consider using GPU-based methods like Victor's TomoQPy, and then maybe have it run on a server that users can access through a web client to process their data. This would alleviate some of the workload that's placed on beamline scientists, and also to have us make better efficient use of our time and everyone else's time. Oops. 1 second. I'd like to thank Olga, Lucy, Francisco De Carlo, Arthur, C, and Barry for their valuable feedback and the development of this project. Also, I'd like to thank Eva and Ken for letting me use their data for this presentation. Thank you. If there's any questions,
thank you, Fabricio. And do we have any quick question? Actually, maybe we should just open the discussion to everybody and all the speakers. Another request, so if everybody is open to the idea, would you please turn on your camera? I just want to take a screenshot for everybody who attended this workshop.
Yeah, and while we're doing that, I also would like to thank user office and event organizers, Connie, Leonine, Jackie, and Kim, for making it happen and assisting us with our difficulties every once in a while with registration and submissions.
I do have a question for Tanya, or yeah, I'm not sure if she's still here.
I'm going to stop the recording. Is that okay? Or
Okay.
ask question? Yes. Okay.